Good morning, everybody. Welcome. Welcome to the 2025 Canada Gairdner Laureate Lectures. Really one of the highlights of Gairdner Science Week and one of the highlights of the year here in downtown Toronto. And before we begin, I'd like to acknowledge that we're gathered on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and we're now sitting on the home of many diverse First Nations, Inuit, and Métis communities. We're very grateful to live and work on this land and to learn from the long history of knowledge and discovery. I'd also like to extend a warm welcome to everyone joining us, not just in the room, but by live stream from across Canada and around the world. I want to especially thank the sponsors who make today's events possible and all of Gairdner Science Week possible. Our particular gratitude to the Kremble Foundation, our presenting sponsor, and as well as the Government of Canada, CIHR, and all of our generous Science Week sponsors for their continued support in celebrating excellence in science. Yesterday's Frontiers in Precision Therapeutics was a great symposium that gave us a glimpse into the extraordinary science that shapes the future of medicine. And today we're going to continue that trajectory with the most highly anticipated talks of the week. No pressure to the laureates. Highly anticipated talks of the week, our Laureate Lectures. Today's lectures promise to give us deep insight into the stories behind this year's award-winning science from the scientists themselves, scientists whose discoveries have reshaped our understanding of biology and disease. Many of them have been traveling across country this week as part of Gairdner Connects, meeting students and researchers, sharing their stories in classrooms, labs, and communities from coast to coast. We're especially pleased to welcome students here this morning because it's your curiosity and enthusiasm that reminds us why sharing science matters. And we hope today's talks will inspire you to pursue your own future scientific discoveries. One of our speakers at the symposium yesterday told us that he was inspired to take up his career by attending the Gairdner Lectures. So it really matters. So welcome to everybody. So thank you all for joining us in what's sure to be an engaging and thought-provoking morning. And now I'm going to step aside and turn over the podium to our first session chair, who is Dr. Bonnie Stevens, Senior Scientist Emeritus, SIGKIS Research Institute, and a longtime colleague of mine. Bonnie.
Good morning, everyone. It's such a pleasure to join you for what promises to be a fascinating morning of scientific discovery and innovation. I'm Bonnie Stevens, and I'm honored to chair this first session of the 2025 Gairdner Laureate Lectures. This morning, we'll explore breakthroughs that span from fundamental biology to digital therapeutics, stories that show how deep curiosity and collaboration can actually change lives. Our first speaker, Dr. Michael Welsh, has spent a lifetime rewriting what we know about cystic fibrosis. His research helped uncover how a single faulty protein disrupts the delicate balance of the lungs, deep understanding that led to new treatments that have transformed the lives of people with cystic fibrosis. Dr. Welsh's work is a model for how careful, persistent investigation can turn biology into hope. Please join me in welcoming Dr. Michael Welsh, who will speak on rewriting the chapter on cystic fibrosis. Dr. Welsh.
Thank you, Dr. Stevens. Thank you, Dr. Rosantin. Thank you for the no pressure comment. So I'm deeply honored to be here, and it's a tremendous honor to be given this talk. These are the words that new parents with a child with CF would face. Imagine the terror. These are the words that started every medical chapter on cystic fibrosis. This has changed. I feel incredibly fortunate to have been able to contribute to the arc that changed this, the arc that started with people with CF, through research, through discovery, and back to people with CF. And that's what I want to tell you a bit about, some part of that arc. So this arc started with Dorothy Anderson. She described the disease and named it in 1938. 10 years later, Paul de Saint-Agnese found that people with CF have salty sweat, and that led to the diagnostic test, the sweat chloride test. And then over the ensuing years, people realized that CF affect many different organs in the body. They're listed here. When you gave someone the diagnosis of CF, though, you were telling them that lung disease, airway infection, inflammation, would be the major cause of morbidity and mortality. And it was appreciated that it was an autosomal recessive disease, an inherited disease. I got interested in this arc because I'm a pulmonary physician, and I was interested in the lung. And I was studying how the lung, how the airways, the trachea, the bronchi, move salt and water across them. I had cystic fibrosis in mind, but I was really just doing this work because I wanted to know how it worked. But then I also got interested because I was in the clinic. I was taking care of people with CF. This is a picture of a lung from someone who had a lung transplant. And you can see all the infection. And I realized that no matter how hard I worked in the clinic, I couldn't change the fundamental course of that disease. So I decided that maybe by combining my research and my clinical care, maybe I could make a difference. There was a lot of work, and then an important paper came out in 1983 by Paul Quinton. And what that said there was that abnormally low chloride permeability in cystic fibrosis. And that was really important. And I began to think about that and began to wonder, is the same thing going on in the lung? So I was able to collaborate with Jonathan Whitcomb from UCSF. He did some of the early studies in developing culture methods. And he cultured tracheal epithelia from a 12-year-old girl who had died with CF. And we studied that. I put microelectrodes into the cells so we could see where the problem was. And we found that cyclic AMP failed to stimulate chloride secretion in CF, and we were able to localize that. It was the apical membrane lacked a chloride conductance. And although we couldn't prove it at the time, the way we were studying that suggested it might be a channel. That began to provide a unifying hypothesis. The sweat gland, the lung, and later the pancreas, the intestine, on down the line, defects in chloride conductance. Really important thing happened when Lap Chi Choi and his colleagues here at the Sick Kids, and Francis Collins and his colleagues at Michigan identified the gene that's mutated in CF, the CFTR. This is an example. I pasted that in the back of my lab notebook. Every lab notebook as I went through had that as the last page, because I was looking at that all the time, trying to figure out what was going on. Then it took about a year, but one Sunday afternoon, out to my house came my postdoc, and she showed me a gel that looked much like this. And there it was on the gel, CFTR. And I said, okay, now we can go. And the first thing we wanted to do was ask if we express CFTR in CF epithelia, would it correct the chloride transport defect? And so we grew cells on a microscope slide. We loaded them with anion-sensitive fluorophore. And if we changed the anion concentration, we could change the brightness of that fluorophore. So here's an example. This was cells that were expressed, we put in CF cells, we put in CFTR with the delta F5 weight mutation. That's the most common mutation. And you look at time 0 and time 1 minute, there's no difference. But if we put in wild type CFTR, you can see those cells lighting up. That was really exciting because it said expressing CFTR in CF epithelia corrects the chloride transport defect. Delta F5 weight did not. So at some level, it's a formal proof of the gene, but more than that, it says you can go forward. And the question we wanted to do is, what does CFTR do? So it has this sort of, is Lapchi here today? Okay. He gave it sort of a clunky name, cystic fibrosis, transmembrane conductance regulator. And the reason they gave it that name is they didn't know what it did, and they're trying to cover all their bases. And the common idea was that it was some type of regulator. It's in a family of pumps. They use ATP to either pump something out of the cell or pump something into the cell. And so that was the idea. That's maybe what CFTR was doing. The other possibility was that it was a channel, that it formed the pathway for chloride to flow across the membrane. Most of the reviews and perspectives and editorials that were written at that time put their money on a regulator, because still today, I think CFTR is the only member of this family that's a channel. So we set out to test this hypothesis, that it's a channel. It's the simplest hypothesis. And as a physician, I know well about Occam's razor. So I followed Occam's razor. And we thought, if CFTR is a channel, then changing the amino acids that line the pore ought to change the selectivity, ought to change the kinds of ions that will go through. So we did an experiment like this. We expressed CFTR in HeLa cells and other cell types. And then we used a patch clamp, patch pipette, and we'd seal it on the cell. And then we could control the solution inside the cell and outside the cell. And we could look at anions flowing across, negatively charged ions flowing across that membrane. And we could look at different anions, chloride, bromide, iodide. And we could do experiments that gave results like this. So here on the x-axis, you see the voltage, on the y-axis, the current. The important point here is where they cross that x-axis. And this shows you that bromide went across this membrane better than chloride, and that was better than iodide. And then we guessed. We didn't know where the pore was in the channel. There were no structures of it. We didn't know what it was, but we made a lot of guesses, and we took a lot of risks, and some of them paid off. And here's how they paid off. If we took a positively charged residue and turned it to negative, we could sometimes get results like this. And this is an example of changing a lysine to a glutamate. And what you can see here is iodide, which had been the least permeable in wild-type CFTR, switched its permeability so that iodide was the most permeable, and then bromide, and then chloride. These data said that CFTR is an anion channel. A year later, Christine Baer and Jack Reardon here at Sick Kids reached the same conclusion using biochemical characterization. So I like this because it connected the genetics, the molecular biology of this channel, and the physiology of the epithelium. We began to understand, okay, now we can understand this defect in chloride permeability. It also said, if you're going hunting for therapeutics, you know what your target is. Your target is ion channel function. The question was, how does CFTR work? I'm not going to show you data. I'll just summarize that. Here's CFTR. It has membrane-spanning parts. Not surprisingly, this forms the pore of the channel. It has something called the R for regulatory domain. It's phosphorylated. That increases the activity. And it has these nucleotide binding domains, highly conserved domains. They control the gating, the opening and closing of the channel. So how do CF mutations disrupt function? What's broken? So I'll show you a schematic to indicate what's broken. So here you have a cell with the CFTR up in the apical membrane where it belongs, the nucleus. There's your genetic defect in the nucleus. And one way you can break CFTR is with a class 1 mutation, defective protein production. Maybe there's a stop codon, abnormal splice site. Protein never gets made. An example of that is the mutation 1282X. Another type of mutation is a class 2 mutation. The protein's made in the endoplasmic reticulum, but as it goes down the assembly line on its way to the Golgi complex, the cell recognizes it as misfolded, pulls it off, and degrades it. So it never gets to the place it needs to do its work. Delta F508 is an example of that. Delta F508 is the most common mutation in people with cystic fibrosis. A third type of mutation is the channel. Goes through the assembly line, gets to the cell surface, it works, but it just doesn't open very often, stays closed almost all the time. G551D is an example of a mutation that belongs in that class. A fourth kind of mutation is it's there, it opens, but it doesn't let anions flow through very well. The pore doesn't work very well. Our example there is R117H. So this mutation classification system told us about ways mutations disrupt CFTR. They informed genotype-phenotype relationships, and they gave a blueprint for developing therapeutics. But does this matter? Does all this knowledge matter? To really matter, you need to answer this question. Can mutated CFTR be corrected? Because then it will matter. So we set out to look at that, looking at delta F508. It's misfolded and fails to traffic to the cell membrane. After we had done that, we knew that quite well, Mitch Drum and Francis Collins wrote a paper that said CFTR delta F508 traffics to the cell membrane, and there it works. And I thought, how could that be? You see this, people publish their results, and you say, what's going on? You know, your first impulse is to say, again, Francis is wrong again. But no, you say, okay, what's the difference in the two experiments? Well, I read the paper, and they did their studies in Xenopus oocytes, frog oocytes. And thinking about that, we had done studies in frog oocytes for a completely different reason. And I thought about that. You keep frog oocytes at 18 degrees centigrade in the lab, whereas mammalian cells are at 37 degrees. If you heat stuff up, it comes apart. If you cool it down, it's more solid. You all know that. If we got dirty clothes, you wash them in hot water, right? Because everything will come apart. So we also knew that protein stability depends on temperature. And so we thought, maybe this is the difference. And so we asked, if we just reduce the incubation temperature, would that repair the CFTR delta F508 defect? Here's an example of what we saw. So this is a gel, shows CFTR, and there's 3 bands. Band A is the immature band. It's not yet been glycosylated. Band B has glycosylation from the ER, from the endoplasmic reticulum. And band C is the mature protein. That's the protein that's fully glycosylated, goes off to the cell surface. That's wild-type CFTR. Here is delta F508 CFTR. You see, it doesn't make band C. It doesn't make the mature protein. It gets as far as the endoplasmic reticulum, and from there it's degraded. And then we just simply lowered the temperature, 30 to 26 to 23 degrees centigrade, and the ratio for fully processed protein goes up. We were so excited to see that. We were even more excited when we were able to measure the current with patch clamp techniques. CFTR delta F508 worked. It got to the cell surface, and it worked. It didn't work super well. There was a problem. Its open state probability, the fraction of time that it was open, was only about 30% of wild-type CFTR. So reducing the incubation temperature rescued trafficking, but channel activity is decreased. So we wanted another way to think about that. And we decided to go after revertant mutations. So we asked, could a second mutation in CFTR delta F508 protein correct the defect? It's called a second site suppressor or a revertant mutation. But you can't really do that. We couldn't do that well in mammalian cells. It just was completely impractical. So we turned to yeast. And we looked at a yeast gene and protein called STI6. It's also, like CFTR, an ABC transporter. And a lot of sequence similarity. It transports peptide A factor out of the cell during mating. And we knew that deleting STI6 causes a sterile phenotype. So we set out to do experiments like this to make a chimera, part CFTR, part ST6. So here in this green line, I show you the NBD1 length of NBD1 in ST6. That, when ST6 has that its own sequence there, mating is 100%. And then we could stick in a part of CFTR, shown here in blue. When we stuck in that part, mating efficiency went down to 12%. I show you the position of phenylalanine 508. And the third one we looked at was the same thing, but missing phenylalanine 508. And the mating efficiency went down almost 99%. I'm only showing you one. We did a lot, took a while to figure this out. But this is the example of the final result we got to. Because then we could just introduce random mutations all over that blue part and screen for mating. And that's easy to do. Here, this is ST6, the wild type CF, a lawn of yeast. Here's ST6 with CFTR, that chimera. Not quite as good, but still a lawn. And here is ST6 CFTR chimera with the delta F508 mutation. The exciting thing is that when we put in a second mutation we found with the R553M plus delta F508, you restored some of the mating. We got the same thing with another variant at exactly the same amino acid, R553Q. This is the negative control. So the revertant mutation corrected CFTR delta F508 function in yeast, so we incorporated it into wild type and mutant CFTR. Traffic improved, but less than it did with low temperature. The interesting thing there was that function improved. Here on the y-axis, I show you a single channel open state probability. That's the fraction of time that the channel is open. There's wild type, about 35% or so. Delta F508, about a third as good. Here's CFTR with the IR553Q mutation. Really didn't affect normal CFTR, but when we combined it with the delta F508, we largely rescued delta F508. So CFTR delta F508 has two defects. It's misfolding with impaired trafficking and decreased activity, and both can be repaired. Low temperature and second site suppressors told us that. And so that provided a roadmap and approaches for developing therapeutics and some confidence to proceed. It said CFTR with the delta F508 mutation is not so badly broken that you can't repair it. We could repair it with two relatively innocuous type of interventions. And then my co-recipient of this award, Dr. Paul Negulescu and his team picked up this challenge. And I'll guarantee you it's a big challenge, but they picked it up and developed highly effective new medicines, fantastic new medicines that have dramatically improved the lives of 85 to 90% of people with CF. I could tell you numbers. I could tell you our adult clinic for CF is now much larger than our pediatric clinic. I could tell you that our inpatient census in the hospital would be 11 to 12 people on the ward at any time, and now it's 0 to 1. I could tell you about the rate of lung transplant plummeting 85%. But even more important were the stories that I would hear from people. People who didn't know if they should go to college or not, because why would it matter? People who couldn't plan for lives. Now they're planning. They're getting married. They're having kids. They're running at our state cross-country meet for somebody who's taken care of people with CF. Watched them struggle and die. It's unfathomable. I cannot imagine this, how good this would be. So how did we get here? It took a lot of people. Here's some of my colleagues, trainees in the lab. We're fortunate. We also have the clinic. And here's the clinic. And one of the important points here is that some of the people are the same in the lab and in the clinic, because a lot of physician scientists, it was so good, had a direct connection with the people, kept our eye on the ball. We also benefited so much from the people with CF. Let me end by telling you about one of them. This is Kay Van Thornhout when she was a little girl. I don't know how old she was there, 5 maybe. And she's using a mist inhaler. Her father was a respiratory therapist, and he made all kinds of devices to try and keep her lungs working. And he did a good job. Kay grew up, and she participated in so many of our studies. These aren't studies just to test a new drug. These are studies designed to help us just understand the disease, and she would participate in those. I remember one time I'm sitting on a wooden bench with her just before the start of a study, and I asked her, "Kay, why are you doing this?" And she told me, "I know this won't help me, but maybe it'll help somebody else." The most noble human spirit. I wanted to cry. Turns out, it did help somebody else. It helped her daughter. Her daughter turned out to have cystic fibrosis. This is a picture of her daughter. She's holding a photo of her mom, and she's smiling. Thank you.
Thank you very much for that really inspiring talk to get us started this morning. So following on the work of Dr. Welsh, we turn from discovery to therapy. As Dr. Welsh mentioned, Dr. Paul Negulescu led the team at Vertex Pharmaceuticals that was the driving force behind the development of the CFTR modulators, medicines that target the root cause of cystic fibrosis rather than its symptoms. The development and clinical application of these medicines is one of the most remarkable success stories in translational medicine. It's a powerful example of how collaboration between academia and industry can bring science to the bedside. Please welcome Dr. Paul Negulescu presenting the Journey to Discover CFTR Modulators for the Treatment of Cystic Fibrosis.
Good morning. Well, you can understand why we wanted to work on this, can't you? So it's just a great pleasure to be here this morning. I'm so grateful for this award, the Canada Gairdner International Award. I said it all the words, and to share it with Dr. Mike Welsh, just a great physician, scientist, and inspiration to me, an example of a humble, focused, generous scientist. I also want to share my appreciation for being able to represent the work of our team. Over 1,000 people have been involved at some point in time in the development and discovery of one of these medicines. And so I'm here speaking for all of them today. This painting behind me is by the first person I met with cystic fibrosis, and he was 10 years old at the time. And he was selling this painting as part of a fundraiser for cystic fibrosis. And so I met him at the fundraiser, and I asked him, what's this painting about? And he said, well, it explains how I feel about cystic fibrosis. The blue is sadness, the red is anger, the yellow is hope, and the green is the sticky phlegm that I'm always coughing up. And I bought that painting, and it is in our research facility right near the entrance, and we look at it every day as a reminder of why we do the work we do. I am an employee of Vertex Pharmaceuticals, proudly own stock in that company, and I'm not going to be speaking about the commercialization of these medicines, and I don't mean to make any representations commercially about how well they work. So it's not a promotional talk, it's a science talk. So as Mike mentioned, cystic fibrosis is a rare genetic disease caused by mutations in the CFTR gene. It was such a privilege for me to share the podium yesterday with Dr. Lapchi Choi and Mike Welsh as we celebrated and talked about that entire arc of discovery from the gene to the medicines. And I really want to start by acknowledging how foundational the discovery of the gene was to our work. And that was in 1989. As Mike mentioned, quickly after that, within a couple of years, work by Mike and other scientists quickly elucidated that the CFTR gene encoded a protein, also called CFTR, which was a chloride channel. And that mutations in that channel affected the function of the chloride channel and impeded the flow of chloride through that channel. And these mutations, as Mike nicely explained, cause different types of defects in the ability of the protein to move chloride. And the one thing that was common about all those defects is that they resulted in cystic fibrosis, which was a progressive disease affecting multiple organs, including the pancreas, the gastrointestinal system, the reproductive system, the skin, and of course, the lung. And ideally, we'd like a treatment that could affect all of those organs and stop the progression or even reverse the disease in those organs. So we began our work around 1999, 10 years after the discovery of the gene. And the reason for that lag is that for the first 10 years after the discovery of the gene, most efforts therapeutically were in the field of gene therapy. We knew what the gene was. Why not just put the normal gene back in the lung or other organs? Sounds really simple, a great idea, hard to do. The lung is actually designed to keep things out, other than air. And so when we try to put genes in, the cells go, I don't think I want that. And so, we're still struggling with that. We're still trying to figure out how to deliver these genes to the lung and other epithelial organs. I think there's great promise for that into the future, but 10 years after the discovery of the gene, organizations like the Cystic Fibrosis Foundation were saying, let's think of another way. Is there another way to solve this problem? And the thought was, well, if we can't fix it at the gene level, maybe we can fix it at the protein level. Maybe a small molecule drug could restore the function of CFTR. And why not go for an oral medication, a pill that someone could take? Therefore, the medicine could go to all the organs affected by cystic fibrosis and restore the function wherever CFTR was in the body. That seemed like an interesting idea, and yet many people weren't working on it. In fact, no one wanted to work on it except our small company was founded on the premise that small molecules maybe can do things that are surprising, and that high-throughput screening, which was a new technique at the time, that took advantage of advances in chemistry, such as combinatorial chemistry, where we were able to create many, many different kinds of molecules of different types, different flavors, different structures, and use automation to screen those molecules for those that might have therapeutic benefit. So we were able to apply that technology to the problem of cystic fibrosis, and we worked with the Cystic Fibrosis Foundation at the very beginning to do that. So we did have a roadmap. As Mike mentioned, we knew a lot about the problem that caused cystic fibrosis, the defects in the CFTR protein. We knew the types of dysfunction those mutations caused. And we also had the relative luxury of one very common mutation that was present in 90% of people with cystic fibrosis, that F508del mutation that Mike mentioned. And so if we could fix that mutation, restore the function of proteins with those mutations, we might be able to affect and help 90% of people with CF. And there were two problems with the cystic fibrosis protein related to the delta F508 mutation. The first was that it was stuck inside the cell, as Mike showed, and I'm showing you that in the graphic here, where the F508 del CFTR is inside the cell. And the second is that the small amount of CFTR protein that gets to the surface doesn't function very well as a channel. So we thought maybe we should try to fix two problems. Maybe we should break the problem into its two pieces and screen for molecules that could either traffic the protein to the cell membrane. We called these protein processing correctors, or C on this graphic. And the idea was those compounds would bind the protein as it's being made inside the cell and help move the protein to the cell surface. And then we could come along with a second molecule called a potentiator of CFTR gating to increase the chloride transport. This would be a second molecule that could bind to CFTR, open the channel, allow the chloride ions to flow out, and of course water would then follow to thin the mucus and help the airway function better. So that was the idea, a combination therapy even from the start of potentiators and correctors. And so we developed assays suitable for high throughput screening to screen for both potentiators and correctors. I'll talk a little about that assay, and I'll talk a little also about our use of human bronchial epithelial cells from CF patients, because that was a critical model to verify that our compounds were working the way we thought. So the basis of the assay was an optical assay, and it was based on technology that was developed by one of my mentors, a fellow named Roger Chen, who won the Nobel Prize for Green Fluorescent Protein, also a Gairdner Award winner. But in addition to GFP, he was also a brilliant chemist, and he developed probes that allowed us to optically measure changes in cell biology, including changes in membrane potential or voltage at the surface of the cell, which proved very helpful in the discovery of ion channel modulators. So in the case of the potentiator assay, we had cells that expressed CFTR, the delta F CFTR, and we took advantage of the observation that Michael Welsh made that room temperature, lowering the temperature of these cells, allows the protein to get to the surface. So without a corrector molecule, we used temperature to correct the CFTR, and then we were able to screen for gating potentiators that could open the channel and make it function better. So we incubated the cells overnight at room temperature to move the CFTR to the cell surface, and then we added test compounds to the wells, thousands of them, hundreds of thousands of them, and screened them individually, looking for a change in color in those cells. And if there was a potentiator in the well that opened the channel, allowed the chloride ions to flow, and it changed the membrane potential. It made that membrane potential more positive inside, and the cells changed color from orange to blue, and that could be detected by a plate reader. In the corrector assay, we just changed the order of operation a little bit. Instead of lowering the temperature, we incubated with compounds, and the few that were correctors were able to move the protein to the cell surface. And then we added a potentiator to open the channel, and then we got the cells to change color again, and that's how we discovered the correctors. So we screened over 1 million compounds over the course of several years, looking for these potentiators and correctors. And they were very rare in these chemical libraries. In fact, our first screen of 250,000 compounds or so in the year 2000 came up with no correctors at all. And we had a lot of folks telling us, well, we told you so. It's pretty hard to fix a defective protein. we thought, well, let's just try some more. Let's get some more compounds. We had a brilliant chemist on our team who did some computational searching for different types of chemicals than the ones we'd already screened. We brought in some more compounds, and we found some potentiators and correctors that became the basis of our drugs. It was very important to screen through these hits, as rare as they were, in secondary assays, control assays, to ensure that we were actually selectively trafficking CFTR and not affecting other processes in the cell that might also affect CFTR, but could be deleterious to other functions of the cell. We were very adamant about selectivity for CFTR, and all our assays were based on trying to find compounds that only affected CFTR function. We left with a handful at the end of years of screening. So now I'm going to tell you a little bit of the journey from those hits to the medications. And this was the brilliant work of our medicinal chemistry team, partnered with our biology team to run this cycle of learning that we call it, where we test compounds, we see how they work in the assay, we develop a hypothesis about what needs to be further improved in the chemistry, and we go around and around and around on a weekly basis to improve the molecules, but also to build into them medicinal properties, making sure that they can be absorbed by the body as a pill, making sure that they have appropriate lifespan inside the body, that they're not metabolized within minutes, and that they can actually get to where they need to go in the cell. They also have to be able to be synthesized on mass scale, tons, quantities have to be made safely and effectively. So these are all the factors that go into the medicinal chemistry. And Sabine Hadida, the leader of our chemistry team, is somewhere here. I see her, there she is, a brilliant chemist and my partner for over 20 years on this journey. And Sabine and her team over the years designed and tested over 30,000 individual compounds in that cycle. Imagine running that cycle 30,000 times, moving toward these more drug-like compounds. And in the end, we came up with a new class of medications that we call CFTR modulators, or CAFTORs for short. CAFTOR has the 4 letters CFTR in it. So if you ever hear about a CAFTOR in your life, you know that's a CFTR modulator. All of them will have that stem as part of their generic name. And over the time of our research so far, we've discovered 5 different CAFTORs that have all become approved medicines. 1 potentiator and a deuterated version of it, as well as 4 correctors, each of which are into 2 different classes, and I'll talk about that. We had to actually find 2 different types of correctors, not just 1. So I'm going to quickly walk you through the process that we went through, sharing with you the hit to drug journey that each of these molecules went on with us. It's like watching kids grow up. They develop, they change, they evolve, they become better in most cases. Not all cases, but most. So in the case of this particular molecule, the hit, for the potentiator, the main challenge was it wasn't very potent coming out of the screen. It was very active, but not very potent. So we needed to make it more potent so that the pill could be small enough to be taken by a person. And Sabine and her team increased the potency of the hit by 300-fold, which was enough to drop it into the 100 milligram range, which is suitable for a drug. You can see on the structure that the left-hand side was pretty much unchanged from the original hit. That's pretty neat that we can use that much of the original hit. But the right-hand side changed dramatically to improve the potency and also improve its bioavailability and metabolic stability in the blood. And just to give you a flavor of what this journey looks like, the team made over 800 compounds in the course of moving from the hit to the lead. And I want to show you the way that this worked, because this was before we had crystal structures of the protein. We didn't actually know exactly where on CFTR this protein, this compound was binding. So we had to almost find our way in the dark, probing the protein with chemistry to find out where the binding site was and to actually get a picture of it. So I'm going to show you how we did that using chemistry as the way to visualize the protein. And so these are all the compounds made in the order they were made. You can see different parts of the molecule being explored. The gray represents areas of protein space that we're reaching into and exploring for the potential of these molecules to interact with CFTR. And you can see that we punched into various different areas, some productive, some not so much, and you can see the potency going up as the movie moves from left to right. Those green lines represent the potency, and higher potencies, higher green bars represent more potent. So it wasn't a linear path. We had some molecules 2/3 of the way through that lost a lot of potency, but we learned from those molecules that they also improved other aspects we were interested in, like the metabolic stability and oral bioavailability. But in the end, we found a great compromise in ivacaftor, which became the first approved medicine to treat cystic fibrosis, and it treated a small percentage of patients for which a potentiator was suitable. Mike described these gating mutations. This drug was approved for those mutations, about 10% of the population. But we didn't want to stop there with 10%, and we continued to work on correctors, which came later in the process. And the reason for that is that a lot more chemistry had to be done to discover these correctors. I'm showing you the hit here on the left, and the final 2 molecules that became approved medicines on the right as first-generation correctors called lumacaftor and tezacaftor, and you can see in the structure how much changed from the beginning to the end. Really, there's only that amide bond in the middle, that NH in the middle, that was preserved, and that is an important interaction, but everything else had to be discovered. And Sabine told me once that she thought we willed these correctors into existence. And I think you can see maybe why she thought that, because if you look at the original hit on the graph there, you can see that not only was it not very potent, but it wasn't very efficacious. It did not move much protein to the cell surface. We used that Western blot assay to determine that, the Western blot that Mike mentioned. So we had to improve both potency and efficacy in this molecule. And this gives me a chance to talk a little about the epithelial cell model that was developed to help qualify these molecules for clinical testing in people. And the amazing thing about the human lung is that you can take cells out of it and they will grow in culture. And with the right culture media, they will recapitulate many aspects of the human airway, including all the different cell types that exist in our trachea and bronchi, the areas that get clogged up with mucus in CF. And you can see in this photo here, which is a confocal micrograph of one of these cultures, the ciliated cells, when the cilia are green, they're stained green. You can also see a CFTR expressing cell in red, and other cells that are helping to make up the surface of that airway. It's quite an interesting ecosystem where all the cells work together. And not all of them contain CFTR. About 1/3 of them do, and those are the 1/3 that generate the chloride transport. So with these assays in place, with these human cells available, we were able to conduct both functional tests, measuring transepithelial chloride transport, and that's shown in the bar graph, as well as confirming the trafficking using the good old Western blot and looking at that B band to C band conversion of immature CFTR to mature CFTR. And what you can see here is that the combination of ivacaftor and lumacaftor in that 3rd bar has more chloride transport than either ivacaftor or lumacaftor alone. So that confirmed to us the importance of the combination for treating patients. And these were cells from a delta F patient. So we took this combination of ivacaftor and lumacaftor into the clinic, and we did see some clinical benefit. We did see improvement in lung function as measured by percent predicted forced expiratory volume in 1 second, or FEV1 for short. And patients on lumacaftor and ivacaftor did show an increase of about 3% in their lung function. They also showed a decrease in sweat chloride, which was an important biomarker of CFTR function in humans. So this was a big step forward for the field because this was the first time we'd shown benefit of a combination in patients with the most common mutation. But it was a modest effect. Patients reported that they weren't feeling worse over time with this medication. It stabilized their function, but it didn't make them feel a lot better. So we knew we needed more function. And you can see why perhaps they didn't respond better, because the chloride transport percent above normal, the percent of normal, was about 20%. So even with both compounds, we were only getting to 20% of a normal lung function, and we wanted to get to 50% or higher to achieve higher efficacy. So we went back to the screening deck, and we screened for additional correctors that would work in the presence of the potentiator and corrector first-generation products. And this was very important because we did find additional correctors, but they were dependent on the presence of the first corrector, and I'll show you that in a moment. So we couldn't have found the second corrector without the first. Everything happened for a reason in a sequence. And you can see in this graph how the triple combination improved function well above the IVA-tezacaftor, in this case, dual combination. So the triple was about 3 times as good as the double in the lab. And I just want to show you a quick video of what this looked like onto the surface cells, onto the epithelial cells under a microscope. So if you can just play the video on the left for a second. These are living human bronchial epithelial cells pre-treatment, before treating with the medication. And there's actually a movie playing. I don't know if you can see it. There's a slight movement there, but little vibration, and that's because these cells are packed with mucus. Mucus is sitting on top of these cells, preventing the cilia from moving, preventing the fluid from coming across the CFTR. And so not much is happening, and this is what the airway looks like in a CF patient. So then we put the same cells, we exposed the same cells to the triple combination for 2 or 3 days, came back and looked under the microscope, and this is what we saw. Yeah, that's what we thought. We thought, woo. It was one of those moments of discovery that I wish every scientist can have. This moment of seeing something for the first time, and you know you've discovered something. You know that this is important. And to have the cells just tell you that directly, nothing fancy, just look under a microscope. There's a value to just looking at things sometimes. And you can see the cilia beating. The mucus has cleared. It has thinned out. This is because the fluid hasn't been able to accumulate on those airway cells due to the increased chloride transport. The fluid has diluted the mucus, which allows the cilia to beat, and this is what we want to happen in people with CF. So we took this triple combination into the clinic, and we saw dramatic benefit as predicted by that airway model. We saw almost a 14% increase in the lung function, remember, compared to a 2 or 3% increase with the dual combination. And you can see on the graph just how dramatic that is. Just to put that in context, a 13.8% increase in percent predicted FEV1, which is the amount of air someone can breathe out in 1 second into a breathing tube, represents about 350 milliliters of air. That's about a Coke can. Patients can feel that amount of air moving through their lungs. They can feel that difference. They report feeling that difference within sometimes the first couple of doses. And this was surprising to us. We did not necessarily expect this much immediate improvement with the addition of these modulators. What turns out, and often these drugs teach you something about the disease, there was a lot of good airway left in these patients. But what was happening is that it was plugged up. The mucus plugs were preventing the air from getting past those obstructions to the good airways. So what this drug was doing was thinning the mucus. Those plugs were going away. Patients would cough them out. Often they'd been in the body for years, so they were black and thick. And the patients would just cough them out, and Some of the clinical trial managers were like, well, this seems like a side effect. This is bad. They're coughing a lot. It's a CF. They shouldn't be coughing. They should be coughing less. But the patient said, no, 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 this is good. I've never coughed like this. It's productive. It's something, I'm getting stuff out. Leave me alone with this medication for a few days. And so this is what happened. They coughed it out, they breathed better, and it was stable. Look at that stability over time. 6 months, they recovered it and they kept it. And then the sweat chloride also dropped dramatically to below the diagnostic threshold of cystic fibrosis. What this means is patients who are on this medication, if they went into the clinic and they did the sweat test, the doctor would say, you don't have CF. That was an incredible moment for those patients because they realized that maybe they're living with the gene, but they won't be living with the disease in the same way. So this was the medication that became Trikafta. So 3 medications in 1 pill, a combination medication. It was 3 pills a day, 2 in the morning, 1 at night. This became a medication that was applicable for about 90% of the population. And Mike mentioned some of the benefits we saw beyond the improved lung function and the sweat chloride drop as we studied the drug in the real world after approval. A dramatic reduction in the number of lung transplants, fewer people in the hospital for pulmonary exacerbations or infections, and reduced mortality to the point where the projections now is someone who starts on a medication like this today at whatever age they are will live to be about 72 years old. Before Trikafta, that was 38. So these sorts of medications have the potential to extend life and quality life by decades. And that's why people are so excited about these advances. So how do the drugs work? I'm just going to spend a minute because, as I mentioned, we were developing these molecules without a picture of what the protein looked like. We did not know where on the protein these drugs bound. They were discovered through cell-based functional assays. So the cells in the protein picked the medications. We did not pick it for the protein. And once the crystal structures could be obtained based on advances in a technique called cryo-electron microscopy, which only have occurred in the last few years, we were able to now see where these molecules actually go. And it was really surprising. The 3 of them all went to a region of the protein that's very far away from where the F508del mutation exists, which I have marked there. They're in a region of the protein that interacts with lipids. You can see the lipid membrane there. This is where the channel sticks into the membrane and where the chloride moves through the membrane. For whatever reason, the drugs went there, and we're still figuring out what the significance of these binding sites are because they might inform us on other ways of tackling these types of protein processing defects. Ivacaftor binds to a hinge region in the pore. You can see it's kind of in the middle of the channel there where the chloride goes through, and it actually twists that channel into a conformation that does not normally exist in nature. It twists it in a way to make it a new type of chloride channel, which was remarkable. The correctors, and you see the one in pink and yellow, they bind to different parts of the protein. And remember I told you that the second corrector needed the first to work. This is explained by the fact that the pink one, tezacaftor, binds to the first membrane transmembrane domain of CFTR. CFTR has 2 membrane spanning domains. And by stabilizing that first domain, it creates substrate for the second one to bind. So they actually work in a sequence in the cell. Fascinating how they work together as a family. And they were designed and discovered as a group, so it makes sense that they leverage each other in their mechanism of action. The other thing that was surprising pleasantly for us was that these molecules not only improved the function and trafficking of delta F, CFTR, but many other CFTR mutations, many that are so rare we would not have been able to study them in patients. And in collaboration with the FDA and other regulatory bodies around the world, we amassed a database that showed that this test, this in vitro test of responsivity, predicted human clinical response. So we got many additional mutations added to the label based on these sorts of findings. And that allows people without the delta F mutation, many of them, to also be eligible for this medication. So this was a very large effort, as I hope this talk conveyed to you. Over 1 million compounds screened, 50,000 or so made, hundreds evaluated in the preclinical phase to advance 15 to clinical studies, 5 of which became approved medicines, the best 5, we think. We conducted over 200 clinical trials to develop the story that I'm able to tell you today. And the real heroes of this are the 11,000 or so people with cystic fibrosis that signed up for the clinical studies to study all 15 of these medicines and advance the 5. Today, we're able to treat about 90% of people with cystic fibrosis, and there are about 75,000 of the estimated 100,000 with cystic fibrosis who have been diagnosed in the world taking one of these medications, only 5 years after they were discovered. So where does this leave us? Where do we go from here? So I hope I convinced you that we use cell-based functional assays to identify chemical starting points for CFTR potentiators and correctors. These are new mechanisms of action that have not been found before, and the high-throughput screening process discovered and taught us about new types of pharmacology. We optimized the hits into drug candidates that were validated in CF airway cells. A lot of work to go from the hit to the drug. We have now triple combinations that can treat about 90% of CF people, extending their lives and improving the quality. But the work is not complete. There are still 10% of people who do not respond to these medications because they don't make any protein. They have a type of mutation that prevents any protein from being made, so the modulators can't do their work. So we need additional medicines, and we're working on that at Vertex, and as well as other companies are working on that to bring CF therapies to everybody. And finally, pharmacological correction of protein processing and function. That's a new type of pharmacology. It's a new type of drug class. And we think it might be relevant for other diseases of protein misfolding or dysfunction. So I show you the 5 molecules here. They're the family. I think of them a little bit like my own family. They all have their own personalities, and they work together like a family should to improve the quality of life. So these are some of the stars, but these are the heroes, the patients. And I'm showing you a few of them here, and Mike told a very poignant story. This lady on the left here, Lindsay, she lives in San Diego. She was actually patient 001 in the first study of a potentiator. She has that G551D mutation, a gating mutation. So she's the person on the planet that has been on one of these modulators the longest, now going on almost 20 years, 18 years or so. And she's running marathons. Before she started taking these medications, she was in her apartment with her cat on an IV drip for the infection she had. And she's just such an inspiration for us. In fact, you can see her real problem is her knee now. It's not her lungs. So that's great that she has other problems to worry about. And then the one in the middle, that's Emily Schaller. She's an older person now. She's in her 40s. And she's helping us all understand how to think about life when you've got more of it to live. And she's helping other people with CF come to terms with having a different type of outlook on life. And then finally, the third, and this is the one I think about all the time, her name is Emily, and she's not responsive to these medications. She's in that last 10%. And she's so happy for the 90, but look at her face. She's like, what about me? And indeed. So I just want to thank everybody who's been involved in this, patients, people with CF and their families, the advocacy group On the right, I show you a wonderful picture of Bob Bell, the president of the Cystic Fibrosis Foundation, with Francis Collins, celebrating the discovery of the gene. And Mike Welsh is there. Roger Chen, my mentor, who discovered the ways to study these types of modulators. And then all the people at Vertex. And that's a little mosaic where everyone at Vertex did a little selfie with a Post-it that had a message for the CF community. And it just, I think, captures the collective effort needed to bring these types of medicines forward. So thank you very much for the privilege of being here.
Thank you, Dr. Negulescu, for that very informative and moving talk. Our next talk really takes us from molecules to people, particularly to children living with chronic pain. I'm very proud to introduce this next individual, who was my PhD student as well as my long-term colleague in the Research Institute at the Hospital for Sick Children and the Bloomberg Faculty of Nursing at the University of Toronto. Dr. Jennifer Stinson has led pioneer work in developing digital health tools that give young patients a voice in their own care. From interactive apps to virtual education, she's reimagining how technology can support empathy, assessment, and management in pediatric pain. Her work shows us that innovation isn't just about new devices or drugs. It's about designing care that truly listens. Please welcome Jennifer Stinson, who will talk on digital therapeutics for pediatric pain, innovations in education assessment and management. Jen.
Thanks, Brian. Thanks, everyone. I want to thank the Gairdner Foundation, and I want to thank the board members who actually were traveling around with us for the last few days. So I can't be in Toronto and not congratulate the Toronto Blue Jays, but the reason I put this up, so we can give a round of applause, is that science is a team sport. And you've heard from my last two colleagues that really the problems we're trying to address today are so wickedly complex that no one discipline, whether it's medicine, nursing, or science, can really address them. We have to all work together to do that. And I'm going to outline how I do that in my lab. So most people think chronic pain is something that only older people get, like myself. But 1 in 5 children and 1 in 5 adults will live with chronic pain. This is costing our Canadian economy $55 billion. And probably 60% of young people that have chronic pain will go on to be adults that have chronic pain, so a lifetime of disability. And my lab is trying to minimize that. We know chronic pain can impact all aspects of a young person's life. This is Cleo Flischer. She was a patient in our chronic pain clinic at Sick Kids, and she drew this artwork for the Canadian Pain Society Art Award, and she won in the theme of pain and mental health. I'm just going to read what she described, this picture is describing. I'm a 13-year-old girl, and I've been living with chronic pain for more than 5 years. I drew this image to express to people how I feel when I'm in pain. It's like I'm drowning. I'm submerged underwater. Pain consumes my whole body. I'm helpless. There's nothing that I can do to stop the suffering or help myself. My lab is about helping these young people like Cleo help herself. In Canada, most recently in 2019, we now have an action plan for pain. So we have 6 priorities where we're trying to focus on to improve the lives of Canadians living with chronic pain. My lab is focusing on 3 of those. 1 is improving access to timely, equitable, and person-centered care. The other thing we're trying to do is increase the education of healthcare professionals. One of my colleagues in the Faculty of Nursing did a study, and it showed that our veterinarians have 5 times more training than healthcare professionals do. So our pets are getting better pain care than our children and adults. And the final part that we're trying to do in our lab is make sure that the digital therapeutics that we develop are really equitable approaches and really get to those communities that are really equity-seeking populations. The other overarching frame that just came out in the last couple years really matches on to what our lab does. We're trying to make pain better. We're trying to make it understood. We're trying to make it visible because it's often invisible. You can look at someone and they don't look like they're in pain. And we're trying to make it better. I wanted to just tell you a little bit about my journey. I'm the first nurse to receive this award. And nurses play a key role, thank you, in our healthcare system. I've talked to so many people over the last few weeks and they're saying, you're the glue in our system. You know patients best. And it's true, we do. But what sparked my interest in science was a grade 11 chemistry teacher. And that's why I have a Bunsen burner over there, because he literally, no pun intended, just ignited this passion in me for curiosity and science. And it was because of a family member that was hospitalized in the ICU that I saw the power that nurses had and the incredible difference that they can make in people's lives. And it changed my course from trying to be a chemist to be a nurse. But I still was really interested in research, and I've been at SickKids my entire career. It's a fabulous organization. I went back to school early on. Within the few months of being at SickKids, my nurse manager said, Jennifer, you ask way too many questions. Why are we doing it like this? Why are we not doing that? And she said, you need to go back to school. And it was true. I went back to school to learn the research methods to address questions that were so important to patients and families. I then went on, I worked in the acute pain service, and that's where I got hooked on pain as a problem and what we could do to try and minimize that. At the time, there was no nurse practitioner training in Canada, so I commuted for 2 years from Toronto to Buffalo to do my nurse practitioner, and then came back to SickKids, worked on the acute pain service again, and then went back to do my PhD with Dr. Bonnie Stevens. And I got a faculty position, and I liked it, but they didn't value what I did as a clinician. And so my dream job came available when Dr. Razant and other people at SickKids created the first nurse clinician scientist position in Canada. This is my dream job. Who goes to work every day and absolutely loves what they do? I do. It's amazing. But I show this to show the young people in the crowd that don't underestimate the power that a simple change in direction in your life can make in terms of where you end up. So I also helped during my PhD to create with Dr. Stephen Brown the chronic pain program in Canada, which is the largest tertiary pain program in Canada, and this are my colleagues, and all my work really stems from there. And this is my lab. My lab is at the back of the room back there. But our whole goal is to try to develop solutions that empower young people to take control of their lives. They know there's not a magic pill that's going to take away, or someday there may be, like CF. But right now, we don't have that for chronic pain. In our lab, what we do is we really focus on this funnel. So digital therapeutics are evidence-based software interventions to use to manage, prevent, or treat medical conditions. And at the core principle of everything we do is that it has to meet the needs of the patients and families. So we co-design everything we do with patients and families in collaboration. We develop them, and then we always have our sight on the end game. We want to implement them. So one of my colleagues, Christine Chambers, we'll go back. She did a study looking at evidence-based digital solutions for pediatric pain and found that 2/3 of them never made it into implementation. That's a lot of research waste. So my lab's all about preventing that and make sure what we develop gets into the hands of patients and families. Dr. Katie Burney was one of my first postdoc fellows, and what we did was a James Lind Alliance priority setting initiative for chronic pain, because as a researcher, I can say, this is what I think we need to develop. But what's more important is, what do patients, families, and clinicians think we should focus on, our priorities? And two of the top things that they told us was, one, improve access to care. Right now, to come see us in our chronic pain clinic, patients can be waiting up to a year to see a specialist, if not longer, in other pain clinics across the country. And what can we do to reduce disparities? And then also, how can we train healthcare professionals to be better providers in terms of pain management? So I'm going to talk a little bit about how we can make pain better. One of the things that Dr. Pat McGraw, who's my postdoc supervisor, and Bonnie Stevens developed was this training network initially funded by CIHR to train the next generation of pediatric pain researchers. So I was a trainee in that program, and now I have the pleasure of overseeing that training program in my lab at SickKids, and we've trained over 400 pediatric pain researchers, and Canada is known because of this, because of the high number of pediatric pain researchers we have. But you can see we're not just training them in Canada, we're training them in developing countries as well. What can we do to make pain better understood? So one of the first applications that I developed was for young people with cancer. So we know that pain is a problem in kids with cancer. At the time that I developed this app, more than 12 years ago, we were moving cancer care from the hospital into the home. But we didn't have really good mechanisms to assess symptoms in a home setting or in a school setting or wherever they were. So I developed this app called Pain Squad, and then I'm going to hopefully show you a little video about it.
This is the Hospital for Sick Children in Toronto. Within these walls every single year, thousands of children are battling cancer and are having to undergo terribly painful treatments. Children like Olivia, who 8 months ago was diagnosed with Ewing sarcoma. The hospital is continually trying to minimize this pain, but to do it, they must first understand it better, and most importantly, discover the medications that are working best. To do this, they need these young patients to record exactly how they're feeling in a pain journal. The challenge is that after multiple chemotherapy and radiation treatments, many kids are too tired or discouraged to even hold a pen, much less keep a detailed journal. And the sad reality is that if this data is not collected daily, it's useless. We needed to find a creative solution to collect data every single day, and with the Pain Squad mobile app, that's just what we did. To begin, our patients were enlisted as recruits in Pain Squad, a special police force dedicated to hunting down pain. We gave each recruit an Apple iPhone loaded with the Pain Squad mobile app. Then twice a day, an alert from headquarters told patients it was time to complete their pain reporting mission. Because the reports worked with iPhone's user-friendly touchscreen, kids could easily fill them out. With a simple flick of the finger, they could identify exactly where and how much it hurt, as well as which medications were working best. But making it easy was, well, the easy part. We knew to be truly successful, we needed to find a way to encourage our young target daily. So we called in some police reinforcements.
Hey, rookie. Welcome to Pain Squad. It's really great you're here. We need all the help we can get to help put pain in its place.
We brought together the casts of Canada's top police dramas, Flashpoint and Rookie Blue, and filmed a series of inspiring videos, then deployed them throughout the app. To encourage the kids to fill in their reports, we built in a graduation structure. When a recruit completed 3 reports in a row, they received a message from HQ informing them that they were moving up the ranks.
You are now officially a full-fledged detective in Pain Squad. Well done.
At this rate, you might even be the next chief.
Don't stop now. You're almost there.
You truly are one of Pain Squad's best and brightest. Keep it up.
Back inside the precinct, recruits could check to see when to fill out their reports and how many badges they had earned. And once their last report was filed, they were sent one final message informing them that they were being retired from the field.
But this case isn't closed yet. Your squad is still fighting. We couldn't have done it without you. So way to go.
The Pain Squad app is now set to roll out into 4 other Canadian hospitals. And due to its success, it will soon be made available everywhere.
The app is an excellent tool. That's her control over the pain because she's able to document it herself, but she's using an app. She just glides right through it, and it makes her feel that she's a part of this. It's fantastic that you all were able to come up with this. It really helps.
Just got the word from Chief. Turns out you've been doing so great, he feels like you're ready to move up. You're well on your way to putting pain in its place. Don't stop now. You're almost there.
Congratulations.
Oh, cool.
Sergeant.
So we then went on to do the studies to make sure that it was reliable, valid, and responsive to pain treatment. It's freely available in the iOS store. But this app was only able to assess pain, and what we really wanted to do was to be able to manage pain. So my first PhD student, Dr. Lindsay Jibb, actually developed Pain Squad Plus. So based on the assessments, it would provide a clinical algorithm on advice to manage the pain. We got funded by CIHR and the Canadian Cancer Society, and we're just completing a multi-centered randomized controlled trial of evaluating this app in children with cancer. So let's look at how my lab is trying to make things better. So one of the things that I hear as a clinician all the time is patients say to me, Jen, I don't know anyone else that has chronic pain. Can you please connect me with someone? And as a clinician, you're often leery about just connecting people without any safeguards in place. So my second postdoc fellow, Dr. Sarah Ahola-Cahoyt, together and myself, we created the iPeer-to-Peer program. So this is a program, well, I'll show you the video and then I'll tell you more about it.
Hey there.
Hi.
I'm Liz.
And I'm Lindsay.
So were you a little nervous for this call?
Yeah, a little.
I'm super nervous, so I won't lie. So I don't know much about you, but I'll tell you a little about me. I'm not exactly sure the type of arthritis you have, but I'm assuming you do have arthritis, right?
Yeah.
Okay, so I have systemic juvenile.
Being a teenager can be stressful. You have a lot to think about, like school, friends, family, hobbies, and everything else. Now imagine being a teen with a chronic or long-term disease. You would also have to think about and manage medical appointments, medications, and therapies, and all when you're not feeling 100%. Learning to be independent and take care of yourself and your health can be complicated. Many young people who are living with a chronic disease have never met another teenager with the same condition. It is easy to feel like nobody around you gets it. So to help, a team of healthcare providers and researchers at the Hospital for Sick Children developed the iPeer-to-Peer program. iPeer-to-Peer uses online video calls to connect young people living with the same disease. Teens are connected with someone a little bit older who has gone through training to be a mentor. This is someone who gets it, someone who has been in the same situation and has learned how to cope.
My name is Shayla, and I live with chronic pain, and I am a mentee.
My name is Carly, and I'm a mentor.
My pain story started in 2010 when I was in a motor vehicle accident. So I have pain from my neck all the way down to my feet. I have nerve problems in my feet. It was really difficult for my friends to understand because obviously they're not going through what I've been going through.
One of the benefits of this program is that it was completely online. So there was no geographical limitation whatsoever. So if you were getting treatment at a hospital anywhere in Canada, or if you were living across the province, or even in different time zones, you could still connect with your mentee because it was all online.
I'd Skype her on my bed, I'd be like this with my phone up here, hey, yeah, I'm not feeling well. But with the Skyping it was really easy because it's I could still feel bad, but that would be the best time to talk to my mentor.
This is a program that I never had the opportunity to experience while I was a patient here at Sick Kids. I wish I had had the opportunity to meet a mentor, someone older who successfully was managing their pain and could learn strategies from them.
She offered me a way how to live with what I'm dealing with, because I know one of my biggest things was being able to accept it. And I ended up telling my mentor that, and she said that the only way for anybody else to understand what we're going through is in order for us to understand it ourselves. Yes, I've had bad days, and yes, I will have bad days and continue to have bad days, but what you do on the good days will overpower the bad days all the time.
This is such a promising program. I can reflect on even one of the mentees that I had that they mentioned that they are looking forward to actually being a mentor one day and hopefully having this experience available to them just because they enjoyed the conversation that we had and the relationship we built so much.
I'd love to be a mentor in the iPeer-to-Peer program because I've seen how it's helped me, so I'd love to give back and like help someone else, like never give up, keep going. Remember where you started off, like it'll be good to be able to get that motivation back going and being able to help someone else.
I actually don't have any other questions. No, it was really fun being your mentor.
It was really fun being your mentee.
So if you have any other questions or anything, you know where to find me. And good luck with everything.
So everything in my lab we do in a phased approach. So we did needs assessment with young people to ask them about what components they would want in this. We did a systematic review of evidence on peer support programs, and we found that programs that provided in-person, more frequent and matched same-aged peers tend to have better results. So we developed a 2-day training curriculum. We had healthcare professionals refer patients to our programs. We've tested this program in youth with chronic pain, arthritis, sickle cell disease, IBD, cancer, congenital heart disease. We actually have a project with sickle cell going on with people in Ireland. And then we have found that this program really improves self-management skills, results in more positive coping, and actually helps people feel prepared to transition on to adult care, which is very scary. But what's most important and promising about this program is that we've already licensed this to not-for-profit organizations who are now carrying this work forward. So Cassie and Friends is an organization in Canada that focuses on young people with arthritis, and they're the ones that are continuing this program for that population. I want to switch quickly into how we can help children with pain in the hospital setting. So about 8 years ago, I started using this robot called Medi in children who were undergoing port access. So they insert a little dome under your skin. There's still a needle that has to go in there for kids to get chemotherapy. It's often distressing for young people. But the robot, we programmed the robot clinically in terms of the steps that would happen. And I'll just show you a little video of Medi.
Pain and anxiety are frequent and significant problems related to cancer treatment procedures for children. Children with cancer often cite needle procedures as the most distressing experience caused by cancer and its treatment. Specifically, the insertion of a needle into a subcutaneous port has shown to be painful, frightening, and distressing to children.
My name is Letty. Please come right on in and sit with me. I work here and get to visit with you.
Based on evidence that we know works to reduce needle pain and anxiety in children, Dr. Stinson at the Hospital for Sick Children decided to investigate the impact of an interactive and friendly robot we called Medi in decreasing the pain from port accesses in 4 to 9-year-olds battling cancer.
I'm going to do some stretching to relax my muscles. You can do it with me.
I'm just feeling. Look, watch Medi. He's here for you.
Sit tight. I'm here for you. Time now for a little clean. It will feel like cool water on your skin. I will be here with you to help you feel brave. Everyone here is very proud of you, including your nurse. Do you have a superpower that makes you so brave? I meet a lot of kids and they showed me something really cool. I'd like to share it with you. It's called belly breathing. You can do it whenever you want to relax. Take a slow breath in through your nose. Hold your breath for 1 or 2 seconds. Now let out the air slowly through your mouth. Wait for 2 seconds. 1,000 to 1,000. Ready? 1, 2, 3, 4. You are being really brave, just like your favorite superhero. Fast girl! 4, 3, 2, 1. Faster! Did you know that when superheroes need to relax, they count to 10 slowly just like we did? Okay, now let's do a bow. You're all done. Woohoo! You did a great job. I'm proud of you.
Medi's interactive vocalizations and movements are key to engaging the brave young patients at Sick Kids in a world away from the distressing procedures they need to undergo to battle cancer.
So the problem with that robot was we built it based on clinical scenarios in terms of what we thought was what was going to happen. But anyone that works with children knows it never goes that way. So I've partnered with engineers in Glasgow to do an AI-enhanced robot. So this robot can actually hear what's going on in the room. It can see what's going on in the room in terms of the child, the nurses, and the healthcare professionals. And so we just completed an RCT, and although we didn't find any significant difference between the group that had Medi versus the control group, you can see there was almost a signal for reducing distress in this population. But what was most important is the patients and family and staff loved this robot. And so now we need to figure out how to implement this in everyday clinical practice in settings for children. I'm just going to quickly talk about another innovation that happened during the pandemic. So in our lab, we have been using VR for children, but during the pandemic, it shut down access to physical therapy treatment. So we partnered with a group at Stanford that developed a program called Fruity Feet. And this VR can actually be triggered by the physical therapist to focus on certain limbs that are painful for the child. And we just completed a pilot trial looking at this in terms of the custom software, off the shelf, and standard care. And this was a trial where no one wanted to be in the control group. So it was like we had to offer, obviously, the VR afterwards, but you can see it was highly acceptable. People felt that it reduced their pain so that they could improve their function, and they were very satisfied with this. The last application that I'm going to talk about is an application that I'm very proud of. It's called I Can Cope, which is a digital platform to help young people better manage their pain, and Dr. Chitra Lalu is the research lead in my lab for this. This is an app where it allows people to do brief check-ins about their pain, and then based on that check-in, it will push them goals that they can set related to sleep, mood, and then based on the goals, it pushes them advice in real time to help them. We've tested this application in arthritis, chronic pain, sickle cell disease. We're just finishing a trial in postoperative pain. We were funded to do a trial in older adults with neurofibromatosis. And I just want to talk about the trial that we completed with young people with sickle cell disease who are a real equity-seeking group and often struggle with access to care. And I mentioned at the beginning, everything I do in our lab, we partner with patients at the center. So Gail and Jermaine were our two patient partners that helped us develop this app and make sure the content was relevant to young people. We then were funded by NIH to do a large-scale randomized controlled trial where they were randomized to the app or an education control group, and then we monitored them 6 months out. We randomized about 60 participants to each group, and in a sickle cell trial, we actually had pretty low dropout because these patients historically have higher dropout rates. But the takeaways were that we really were able to recruit people in this, and that the young people completed the outcomes at 3 and 6 months, and that we found significant improvements. So these young people not only have acute pain from vaso-occlusive crisis, but they have chronic pain. And so we found that they had less days with pain and less painful episodes. They had reductions in, or improvements in their mood out to 6 months. And there was some change in terms of reduced in activity limitations and interference with pain. These are just some quotes that young people said. If I didn't have a good day, I would mark it down. It would encourage me to try harder and have positive energy. If I didn't have a good sleep the night before, I would put in my little score, and then it would encourage me to go to sleep earlier or try other strategies. We now got funded by CIHR to do what's called an implementation trial. So we're implementing this app in the sickle cell clinics across Canada, where we're doing a preparatory phase, trying to find out how the clinic staff will prescribe this to patients, what might some of the barriers be, what might some of the facilitators do. We'll implement it, and then we'll develop a roadmap to scale this up. The last intervention I want to talk about, and again, it speaks to researchers being nimble during the pandemic. Again, I talked about how the pandemic shut down all the chronic pain programs in Canada, so I was funded by CIHR to develop a one-stop shop where young people could get evidence-based strategies. So our I Can Cope app's in that. It was co-designed by young people, and we have 2 trials underway right now. So just to wrap up, I think hopefully I've convinced you that technology is a way forward with young people. Some of the things that we're working on in the future are how we can integrate better AI components. So we're integrating a chatbot into the sickle cell program. And we know it's going to be challenging to implement digital technologies because we do have fragmented healthcare system. There's inequality in access to care and limited system integration in the hospital settings. And we also want to make sure that we work with policymakers to ensure we can develop the infrastructure to implement these more rapidly in the hospital setting. So again, closing thoughts, it's important that we use equitable inclusion of young people. Their voices are critical. I think you've heard all the speakers about how it's important to listen to and learn from patients. I'm really committed to training the next generation of pediatric pain researchers, so I'm hoping in the future to see another pain scientist up here, another nurse on the podium, and that digital interventions really can successfully empower young people to improve access to the care that they need. And new technologies such as AI will enable us to enhance therapeutics, but we need to make sure that we put the guardrails on, especially around AI, ensuring patient safety. So with that, I just want to thank you for listening.
So thank you to Jennifer Stinson, and thank you to all of our speakers this morning. It's been a very exciting first session. And now it's time for a break. I think we're running a few minutes late, but we'll still take the 15-minute break. 10. I'm looking at the boss down here. 10-minute break. So we'll welcome you back shortly. Thank you.
Say you like the wind blowing through your hair. Well, come on, roll with me till the sun goes down. Texas sun. Say you wanna hit the highway while the engine roars. Well, come on, roll with me till the sun goes down. Texas sun, oh yeah. Texas sun, caressing you from Fort Worth to Amarillo. Well, come on, roll with me till the sun dips low. Texas sun. Texas sun, oh girl. Texas sun. When I'm far from home and the cold winds blow, stuck out somewhere, the folks I don't know. 'Cause you keep me nice and you keep me warm. Wanna feel you on me, can't wait to get back there again. Texas sun. Texas sun. Texas sun. Texas sun. You say you like the wind blowing through your hair. Well, come on, roll with me till the sun goes down. Texas sun. Texas sun. Oh, baby, you're so gorgeous. How about you and me? Take a little trip. In the big body. Take a ride with me, babe. You by my side. How does it sound? You and I. Oh, girl. Take a ride with me, babe. You by my side. How does it sound? You and I. Baby. strong, and the love is tall. Ain't got no name, ain't got no fancy education, but I can see right through a powdered face on a painted fool. Let me slip through. Why you tryna hold me back? I'm just tryna move up front, little more this, little less of that. Can you feel me? Let me come through. I'm tired of being in the back. I'm just tryna move up front, little more this, little less of that, yeah. They tell me I was born to lose, but I made a good thing out of bad news.
Okay, can I encourage everybody to please take their seats for the next exciting session. It's great to see so many people here and so many good conversations going on in the room, and seeing people I haven't seen for a very long time, Peter. So welcome everybody back to our next session. We still have some amazing people to hear from during this next session, but I'm going to again hand over the chair microphone to Kyle Brown from Moderna, Canada. So Kyle, please come and take over.
Welcome back to the 2025 Gairdner Laureate Lectures. As was mentioned, my name is Kyle Brown, Associate Director and Field Medical Advisor for Moderna, Canada. It's my pleasure and my privilege to be able to chair our next session for you all here today. This half of the program takes us across continents and disciplines, from global health and nutrition to cancer epigenetics and fundamental genetics, all united by a common thread: curiosity that leads to impact. We begin with a scientist whose work has improved and in many cases saved the lives of millions of children around the world. Dr. André Briend transformed the treatment of severe childhood malnutrition by developing ready-to-use therapeutic foods, simple, scalable interventions that reach the most vulnerable communities. Please welcome Dr. André Briend speaking on changing the way we treat severe malnutrition.
Thank you, Mr. Chairman. Before starting, I want to say this is a great honor for me to receive this award. But I want as well to say that all the people who work with me to improve the treatment of severe acute malnutrition should be associated with this honor, and also all the people who are currently working in the field, very often in difficult conditions. I want them to receive also part of this honor. So changing the way we treat severe malnutrition. What is severe malnutrition? WHO in 2009 published an official definition based either on a low weight for height index, which is something you calculate weight after measuring weight and height and putting that in a table, or low mid-upper arm circumference, or presence of edema. We don't know how many children suffer from this condition every year, but it's more than 20 million children. Severe malnutrition is associated with a high risk of death, and up to 2007, WHO recommended to treat children with severe acute malnutrition in hospital. On the bottom part of the slide, you can see the association between mid-upper arm circumference or MUAC and the risk of death, and you can see it increases dramatically. On the picture, you can see one of these children, and you can see how you measure the MUAC with a special tape with a different color appearing on the window when the child is severely malnourished. So currently, more and more people assess malnutrition by measuring arm circumference, and there is a long history behind that. The idea was first proposed in the 1960s to measure nutritional status of children in places where you had no scales, because the traditional pediatric practice is to measure nutritional status by assessing weight and age and height. So it was a kind of surrogate measure in places where in very poor places. But in the 80s, there were several people who asked the question, among all the nutritional indices which are on the table, which ones are the most effective to identify children with a high risk of death? So several groups work on that, and to our surprise, I was part of a team working on that. To our surprise, we found that middle-upper-arm circumference, which is easy to measure, is very effective in identifying children with a high risk of death. And this is a paper we published in The Lancet in 1987, as far as I remember, which just showed that. Also, this study showed that the number of children with a high risk of death is too high to be treated in hospital, as was recommended by WHO. And in addition to the issue of number, there was also a problem of social acceptability. In Bangladesh, in a rural conservative society, having women moving to the hospital to accompany their children was something which was difficult to accept. And in Senegal, where I used to work before, in rural community engaged in hard agricultural work, having the mother being away for a month was a big problem, and very often family refused referral. The reason why WHO recommended the treatment in hospital is what the basic treatment of the children was to feed them with liquid-based, milk-based diet. And here you have the recipe which was recommended by WHO. There are two recipes, F75 and F100. And let's look at the F100 recipe, which is used for a greater part of treatment. You can see it is made by mixing together dry skim milk, oil, and sugar. There is a lot of oil in this preparation, which is a problem because when you mix all these ingredients together, oils comes on top. So you have to mix the ingredient frequently. Also, there is a big problem is that if you accidentally contaminate these feeds, bacteria grow exponentially in it, especially when temperature is high. So this kind of diet is unsafe to use at home if you have no perfect hygiene, no refrigeration, and so on. Although this diet was complicated to prepare because you needed to add to it a mixture of mineral and vitamin, which you have the recipe down on the bottom of the slide, which was rather complicated. The risk of bacterial proliferation in case of accidental contamination was due to the presence of water. And when I was working in Bangladesh and doing this study on the association between anthropometry and mortality, I was sharing my office with a nutritionist from Guyana, who was working on examining which foods are most heavily contaminated in rural Bangladesh. At that time, I was working in ICDDRB, which stands for International Center for Diarrheal Disease Research. So the question of hygiene was very much receiving attention. We were sharing office and, of course, discussing together about our findings. And he told me that his research could boil down to a simple observation. Foods which contain water are dangerous, especially at high temperature. And so this is a table taken from one of his papers, which we just see that food containing water have a higher number of bacteria after a few hours, and the difference is huge. It can be increased, the contamination, by a factor of 100 or 1,000. In 1993, there was a big change in the management of severe malnutrition. A gentleman called Michel Lescan started a small company to industrially prepare the F100 formula recommended by WHO. So this was a huge project because there was no more error in recipes, so all the ingredients were prepared together. And also he used a trick that I will explain in a minute, to avoid stirring. That is, he made sure that the fat component remained in suspension after adding water in it. So this was a huge project, but remained the problem of risk of bacterial proliferation in case of accidental contamination. So for that reason, it was used in a kind of temporary inpatient setting, mainly in emergency settings. So when I saw the success of this food, and I had in my mind that what we need to treat the children is dry food. I want to see this Michel, and I told him, why don't we prepare a food which is high fat and which can be consumed without addition of water? So initially we looked around, examined several recipes, pancakes, donuts, biscuits. We spent some time with biscuits because biscuit was already used in relief operation. The problem with biscuit, well, the problem with all this food, but especially biscuit, is that the vitamins you put in these feeds are destroyed by heating. And for preparing biscuit, you need baking at a very high temperature, 180 degrees Celsius. And also we had a problem with biscuit because, you know, the WHO diet, which was recommended, was very high in fat. And if you want to prepare high-fat biscuit, you get very fragile biscuits which crumble very easily, which was not easy to transport. So at the time we thought, well, all these are problems. Why don't we try solid bars, a little bit like the bar that you athletes or cyclists use during competition? So we spent some time examining that. But again, we face problem. We face problem because the problem was in the choice of fat component. The secret of good food is, fat food, is to have a melting temperature around 37 degrees. Because in that case, it melts when you put it in your mouth, and it absorbs heat, and it gives a very pleasant taste, a feeling. But the problem is that a fat bar, which would be soft above 30 degrees, it's not possible to use in tropical climates. So we have to abandon the idea. So the alternative was to use the high fat, fat with a high melting point, but in that case the taste was awful. So then we thought, well, these soft bars, why don't we turn them into a spread? And apparently you recognize a very popular spread which is used in France and very much loved by children. And I looked at the composition and I found it was very close in terms of ratio of protein, carbohydrate and fat to what was recommended by WHO. And also, this is something I did not realize, but spreads do not contain water. So it's interesting. And also, during preparation, there is no requirement for cooking, for baking. So it looked very interesting. And at this stage, I should tell you the difference between a powder and a spread. So I told you that Michel prepared a powder feed, which did not, with fat which did not come on top of the preparation and which did not require constant stirring. And the trick was obtained by using small fat droplet around which they put water-soluble vitamins, water-soluble ingredients. So in that case, it remains in suspension. And the spread is the opposite. It's fat, and it's fat with all the soluble components which are inside the fat. And actually, some foods, if they are fat enough, they can be presented either way. It's a kind of what we call in chemistry phase inversion. So the idea looked attractive. And what Michel proposed is to replace part of the dry skim milk entering in the WHO formula by peanut butter, which has the right melting temperature. And also it has a pleasant taste. And also it has another advantage, is that the vitamin and mineral that we put into, I found that has an awful taste. It contains potassium chloride. I don't know if you had a chance to test it, but it's very salty. But once it is in the peanut butter, I mean, peanut butter with a salty taste is fine. So spread preparation is relatively simple. So it requires no cooking, just mixing. You have to increase slightly the temperature to get the proper mixing. So, but up to 40 degrees enough, so you don't damage the vitamin. So just by replacing part of the dry skim milk by peanut butter, you obtain what we call ready-to-use therapeutic food. So then I wanted to check that really bacteria do not grow in it. So I had a hard time to convince a colleague, a bacteriologist, to do an experiment, because he told me if there is no water in your stuff, of course bacteria won't grow. We all know that. But anyway, we did the experiment, and we found that bacteria did not grow in the spread, in contrast to the liquid feed. And this graph gives the bacterial count with the log scale. You can see, what I said before, in a humid temperature, when you mix food with water, the bacterial count increased by a factor of 100. So these foods are safe to use as home, even in absence of refrigerator. Apparently we're stuck to this one. Okay. So then the next step was to see whether children would accept it. And then we faced a hard time because nobody believed in the idea. Everybody said that children with severe malnutrition should be fed with liquid food. We have been doing that for many years. Why should we change? So it took a long time to find an NGO who was ready to accept to change one feed. Children who have severe acute malnutrition, they are fed 6 to 8 times a day. But people thought it was too risky to replace all the feed in the first trial, so we replaced only one feed. And what we found, we took each child with his own control, and we found that children preferred the solid food to the liquid food. So again, we were very surprised, but when I discussed with colleagues working with test physiology, they just told me that they discovered the principal idea behind the most birthday cakes. If you want to have something very tasty, you need to have a high energy content, because naturally we are attracted towards high energy density foods. So you don't have a good birthday cake with less than 500 kilocalories per 100 grams and a lot of fat and a lot of sugar. So we just confirmed that. And then the next step was you should try this new food in the context of, replacing all the F100 with this solid food. And then I have to thank and pay tribute to all the group of the university, Sheikh Anta Diop University in Dhaka, who did a study with you have the picture of Issakha Diop, the PI, and they did that. They treated randomly assigning one group of children with the classical liquid food and one group with the high energy, the spread. And they found that the weight gain was superior. And then the next step was to move to the community, and it was something difficult. And then here I have to pay tribute to another colleague, Steve Collins. And he worked in a very special condition. He was in the years 2000, around 2000, he was in Ethiopia, where there was a food crisis in rural Ethiopia. And apparently at that time, Ethiopia was taking up on economic aspect, and the government was not very keen in opening therapeutic feeding center to have a big crowd of children with severe malnutrition. They wanted to move away from the image of Ethiopia being the country of famine. And then Steve Collins said, well, but I heard there are guys in France who developed a food which can be consumed at home. Why don't we try that? And the government said, yes, you can have a go. And Steve and his group, they treated 100 children just like that in Ethiopia, and they found it worked very well. And they were so enthusiastic about it that they wrote a paper in the Lancet arguing that we should change the way we treat severe malnutrition in famine. So I think it was a good achievement, because it's not easy to publish a paper in the Lancet, especially when you just report anecdotal treatment. Anecdotal report of treatment without any comparison group. But apparently it was thought to be revolutionary enough to grant to be published. There is another major contributor on also moving to community is Mark Manary, who used to work and still work at St. Louis, Washington University. And at that time, he was working in Malawi. I was based in Paris, and I was desperate to find people who would be ready to treat the children with severe acute malnutrition at home, because this was the ultimate objective. And most people were reluctant. And then an American colleague visited our lab and said, you should get in touch with this guy. So first, we did not know each other. We just corresponded by email. And he started the trial, and the rationale for him to start the trial is that he found that the traditional hospital-based treatment had limitation and didn't work very well. And he did a comparison trial and found again that treatment at home was working. So following that, the news spread around, and quite a few NGOs started to treat children in the community, and they found that RUTF can be safely used to treat children at the community level, and children without complication can be treated right away, and they represent 80 to 90% of children turning up for program. And children who do need treatment because they have complication, usually they stay 5 to 10 days, not long, compared to at least 1 month before on full treatment in hospital. So when you put all these figures together, more children you are able to treat right away from the beginning in the community. And those who stay in hospital, they stay much shorter. You increase dramatically the capacity, the number of children to treat children. So as a result, in NGO, I told you initially, many of them were reluctant to treat children in the community. They felt safer to treat them at home. But there were pressure coming up from the field staff. They discovered that the NGO next door who were treating in the community, they treated more children and they got good result. So all this led the WHO in 2007 to make a joint statement with UNICEF and WFP. And they introduced for the first time the use of MUAC in the definition of severely acute malnutrition, and also they recommended, they endorsed the treatment with RUTF. So now UNICEF is starting to use that extensively, and it describes the food as a wonderful. And in 2023, RUTF was included in the WHO model list of essential medicine. Now there is a continuous increase in the use of RUTF since the early 2000s. We don't have a statistic before 2005 because the number were too small to be, so that nobody took care of calculating how many children were treated with severe acute malnutrition. Most of them were treated by NGOs, and a few of them were treated in dedicated pediatricians working in hospitals and who had a special interest in malnutrition. So it's a huge project, huge progress. So in 2024, the UNICEF published a report in which they say, we estimated that in 2023, 1.2 million deaths of children aged 6 to 59 months were averted by programs that ensure timely access to CMAM services. So, at this stage, I should tell you that I am a medical doctor, but in addition to that, I am trained as a scientist. And as a scientist, I am trained to believe only in data which are published in peer-reviewed journal, and even then, only after careful checking. So actually, I am asked very often, how many lives you think you have saved with this product? Yeah, the honest answer I say that I don't know. Because if you really want to know, you need to treat one group of children and another group of untreated children and control. Repeat the experiment several times and then do a meta-analysis, which is clearly impossible. So I don't know. And if people insist, I say presumably maybe, presumably many, and I stick to this answer, which I think is compatible with that. But I have no 100% confidence in this figure. So I learned several lessons during this RUTF story. First, that innovation comes very often by connecting very well-known fact. All bacteriologists knew that bacteria need water to grow. All food technology knew that spread contains no water, they are fat, and they are prepared without baking. And I knew that children with severe acute malnutrition, they need a food which is high fat and with vitamin and mineral. So connecting all this together, it was simple, the solution, quite kind of obvious. So very often I am asked, what advice would you give to young colleagues? So my first advice would be talk to colleagues, especially if they work in different disciplines. Second lesson I learned from this story is that innovation meets resistance. And here I like to quote a sentence which you find very often in articles on innovation, which is much of new knowledge is certain to arouse opposition because of its tendency to challenge current belief in practice. And this is a sentence which was pronounced by Vannevar Bush, which was advisor to President Roosevelt during World War II. In our case, we face strong resistance. I don't want to go into too many details to that, but leading experts said it won't work. It won't work. It's a bad idea. So my second advice to young scientists is don't trust too much the experts. You should listen to them. Sometimes they say things which are worth considering, but sometimes they are wrong. So there were many reasons why people were reluctant. They were afraid of higher osmolarity. They confused the product with condensed milk. Some were afraid of excess renal solute fluid, which could lead to hyperosmolar dehydration. But actually, this is something you observe in children who are fed bottles with too concentrated powder, but it doesn't apply to children who have free access to water. Of course, there was the fear of peanut allergy, but as a reminder, peanut allergy is something you don't see or very rarely in Africa because children are exposed to the allergen. And also there was a fear as well of having the child not at constant supervision, clinical supervision. But people also forgot that just putting together as a big crowd children who are often infected in a small room in imperfect hygienic condition is a recipe for cross-contamination. And you know that even in the most advanced country, I'm sure it's the case in France, I'm sure it's the case in Canada, hospital ward is a place where you get infection, whatever you do. So sending the children back home as soon as it is not necessary to keep them is the good idea. Another lesson I learned also, which is very sad and it's very much on the news, is that progress can be reversed. A lot of the program treating severe acute malnutrition used to be funded by USAID. And it's very sad to see that some programs are completely damaged for that. So you know what happened with the USAID. For me, it's very sad because from the early program, quite a few were funded by USAID. They had bright people over there who understood the potential, I would say even before WHO, and who are keen moving that. So I feel very sad of seeing this institution in which you had one of quite a few of the brightest nutritionists of the world, which is now, this expertise is neglected. And also this cut in funding is observed in other donors. European Union is under threat now and expanding its military expenses, so it's competing with it. So we need for alternative funding, and in that case, I want to mention an initiative of 3 French NGOs who advocate for using part of the soda tax, which is a tax which is introduced to discourage poor people to eat or drink, sweet food. And they suggest that some part of this tax should be used to purchase this kind of this UTF fund program. And this could be done both in rich and in poor country, because you can go to any village in Africa, you will find a shop with a fridge working with solar power or gas and selling sweet drinks to poor people. So it's complicated because in Europe we introduce sugar, tax on sweet beverage, but also we subsidize beet grower producer for sugar production, so it's complicated. And also, there's another concern which means that the progress can be reversed, is that there's a threat now with the climate change. UNICEF just published a report about the implication of climate change on child nutrition, and it's rather frightening. Yesterday I read on the news that coal production reached record level last year, and so this is a big concern. We are not on track to limit the increase in temperature below the 1.5 degree target. And final word is that although I have spent my life trying to improve the treatment of severe malnutrition, I'm a big supporter of prevention, and I think prevention is much better than cure. But unfortunately, we have to face the reality. There was a report by WHO published in 2020 explaining that the costs of healthy diet exceed food expenditure in most countries in global foods. These diets are unaffordable for over 50% of the population of sub-Saharan Africa and so on. So this is a major concern. I thank you for your attention.
Thank you, Dr. Briend. The Gairdner Early Career Investigator Program honors exceptional career researchers across Canada who are already shaping the future of science and health. Dr. Prativa Baral is building health systems ready for whatever comes next: pandemics, disasters, and emergencies. Through her research at McGill University, she's working to ensure that evidence and readiness go hand in hand. Please welcome Dr. Prativa Baral, presenting Faster, Smarter, Stronger, Building Health Systems Ready for the Next Emergency.
Hello, good afternoon. Thank you so much for this honor. It's such a privilege to be here and to speak right after hearing about the incredible work that so many of the laureates have done. And a special thank you to Dr. André Briend as well, whose work has inspired so many of us in global health. Oh. Okay. I'm just going to awkwardly stand here until, oh, here we are. Okay. So I'm going to start with a quote, and I think it'll land really well in this audience because it's a quote by a science philosopher. And basically the quote is one that I've been thinking about a lot. It's followed me through every health emergency that I've worked in, whether it's COVID, whether it's climate shocks. And it's the idea that when systems collapse, the question that everybody asks is, what's happening? And often we just simply don't know. So all of our knowledge is only some kind of approximation, and the approximation can be improved. We do not know, we can only guess. And my work has been about improving guesses during health emergencies. And rethinking health system readiness in the context of health emergencies so that health systems are faster through better data and evidence, they're smarter through better systems and workforce, and they're stronger through better trust and communications. So 6 months into my PhD, the pandemic hit. And so my PhD years were very much all about how do we make health systems faster. The answer to that, which might sound very simplistic because it's a lot more complicated than that, is that we need data and we need it in real time. The problem is traditional data cycles are slow, and in an emergency, you want the wheel that you're seeing on this slide to move faster and faster. And instead, the opposite tends to happen. By the time the right information has reached the right decision maker, the situation has already changed, and we saw that over and over again during COVID. Or you're collecting data once every 5 to 6 years with health facility surveys, for example, and they're great, they're comprehensive, but they're not really helpful during health emergencies. And so what happens then is throughout the pandemic, that's one of the things that I worked on. I worked with the World Bank and the GFF and partner countries around the world to try to find ways to improve this data cycle so that it was moving fast enough to guide decision making. As you'll recall, we were trying to figure out how to allocate resources accordingly, which regions were being most affected first, what was happening, and where do we act first. And so this is an example of some of that work. The graph shows country data, and because of data sensitivity issues, I'm not saying which country it is, but it's not Canada. And essentially what we were doing is we were trying to monitor how essential health services were being disrupted as a result of COVID. So on this graph, on the y-axis, what you're seeing is the average number of outpatient consultations per facility, and then on the x-axis, you're seeing time. So we went to countries and we said, okay, what data do you have that's trickling in to kind of get a sense of what's happening, what kind of service disruption is happening during COVID? And they had administrative data. So those are health systems data from dozens of countries, and we projected what we thought was going to happen based on historical trends, based on seasonality, and then in red, what you see is what actually happened. And as you can see here on the right square, what actually happened is that there was a 20% reduction, more than 20% reduction in service utilization in the peak phase of COVID. And this points to problem one, because most countries don't have reliable monthly data, especially during emergencies. And so as a result, more often than not, you're extrapolating. So let's say you're going in February 2019 and you're taking a snapshot of that particular period of time. And because you don't have data, you're kind of extrapolating to the future. And that's a problem, as you can see, during health emergencies. So we did this for a number of different countries around the world, for a number of different important indicators. And if we look at OPD, this is a paper that we published at PLOS Medicine. OPD, so outpatient consultations, the graph on the very left, this shows the average of the disruption in the partner countries that we were working with. So on average, what we saw was that there was a 13% drop in outpatient visits. And if we were to model that, that translates to over 113,000 excess maternal and child deaths during the pandemic. And so these numbers, the fact that there's a data lag, aren't just statistics. They're evidence of what delayed information costs. Because had we known that these services were being disrupted in real time, then maybe we could have done something about it to avoid service disruptions and excess deaths. So problem two, I kind of alluded to that earlier, is that even if you have the data, if the data is incomplete, then that's not helpful either. You have biases, you're making assumptions. And this is another country that I was working with during COVID, and we can see that there are a lot of indicators that are either always unavailable with a massive spike in the lack of completeness in the COVID phase. So that's the blue graphs that you're seeing there. So my team and I wanted to do something about it. We realized that routine data can only go so far, and this is particularly true during an emergency like COVID, and yet countries still had to make decisions. And so we created a rapid data analytics system, what we conveniently called FASTER, or the Frequent Assessment and Systems Tools for Resilience, so that the wheel that I showed you earlier can move faster and faster. And so the idea with FASTER was to create a methodology that provides real-time pulse checks of what's happening on the ground on disruptions between traditional forms of data collection. And because we created this methodology during COVID, it was short, it was nimble, the questions that we were asked were adapted based on the country's needs and priorities at that time. And they were phone-based, which was very much a game changer at that point, because it meant we didn't have to go to health systems. We could just do it via the phone. So we collected data frequently, we analyzed it in a timely fashion, where we also automated a lot of the analysis, because we were doing this on a monthly basis to a lot of countries. And then we shared that with country partners, who would then figure out which health system gaps to prioritize. And then they would tell us what else they wanted and what else to investigate. And then we did this over and over again during the pandemic. And because of the smaller, constant, shorter pulse checks, we're not just receiving one snapshot in time, we're really measuring not just readiness, but also resilience over time, and really getting a sense of the trends of whether the health systems were absorbing the shocks or which health services were being disrupted. And the system, this methodology, is now being used in over 18 countries and more to monitor health systems and inform decision making. So I'm going to give you a specific example of the trend assessment. So this was some work that we did with a couple of countries, also in the acute phase of the pandemic, when vaccines were about to be available. And a lot of the government partners were concerned about vaccine hesitancy, and rightfully so. And so we were able to track the evolution of vaccine hesitancy across several countries. And what you see on the left are just the pulse checks that we did and the countries that we looked at. And on the right, because we were able to monitor it rapidly and quickly and frequently, we actually noticed that it was actually not a question of hesitancy, it was really a question of access, at least among healthcare workers, which seems very obvious to us in hindsight, but at that point in time, had we not monitored it as frequently, we wouldn't have known this as quickly, and this has massive implications, as you can imagine, for policy. So this is a small segment of some of the questions that we added and adapted. And this is to say that data is political, right? Health crises are political, and it's hard to measure during emergencies. It's really hard to do that well. And this is just to give you an example of additional questions, additional modules that countries had, because we were iterating this tool over and over again. And so whether it was vaccine hesitancy, whether it was questions about PPE, whether it was questions about childhood immunizations, we were able to add questions as needed based on the country's political reality, country's needs, and scale it across all of these countries during an emergency. So it was really interesting and stressful to iterate a tool and adapt it during an emergency and to scale it and find a way to balance core standardized questions so that we can still compare, while also recognizing that there were some very country-specific needs that we needed to address. And so these rapid cycle assessments are really helpful because they capture change in the health system, but also capture country context, which is really, really important during health emergencies. And FASTER, as I've mentioned, is now used not just for COVID, but to measure all sorts of shocks from violence to shortages in medicines or natural disasters. I'm going to briefly show you a small other example that's more Canada specific. So I'm a huge data nerd, as you can imagine. And so I firmly believe in the idea that better data, more frequent data is what we need. So this is an example of a project I was involved in with the COVID Immunity Task Force right here in Canada, where we were trying to build a network that was built on residual blood samples so that we can assess emerging and endemic pathogens that are emerging. And I know I'm running out of time, so I'll quickly tell you a little bit about some of the work that I'm doing as a new faculty at McGill. I joined in May, so very new. But this is this recognition that even if you have the best information and the best data, if you do not have systems that can act on the data, then it's not really helpful. And so we are thinking about this in the context of changing climate, and frequent climate shocks, as well as Canada's senior population outlook that has uncharted territory. So in that context, where our health systems right here domestically will be quite burdened, how do we help? And so this is a new project that we've just launched, and it's in response to the realization that the current emergency model is quite reactive, it's military dependent, it's unsustainable, and we saw that during COVID. And so we're building a future model of a civilian reserve that's coordinated, local-led. It's basically a surge system that would act to the data that's being collected, that's embedded in communities, and that would be ready to respond to across crises from wildfires to pandemics. And we have proof that this model can work. We have certain specific groups that have popped up right here in Canada. And we're working with a couple of partners from across the country and having initial conversations with the Red Cross to design the blueprint of this emergency reserve, looking at governance options, financing models, and workforce strategies so that once we have the data, we also have a responsive mechanism to act on the data. And then the final thing that I'll say is you can have data, you can have smart systems that can respond with a trained workforce, but systems fail when trust fails. And that's basically what we're trying to, one of the pillars that we're trying to navigate at the Pandemic and Emergency Readiness Lab at McGill. I'm co-leading that lab with the former international president of MSF, Dr. Joanne Liu, and we're essentially a research lab that's trying to strengthen health systems readiness with the data piece, the ones that I mentioned, with the workforce piece, but also the trust piece. And we're hoping to put all of this together in order to make sure that our health systems are faster, smarter, and stronger in preparation for the next health emergency, right here domestically, but also globally. Thank you very much.
Thank you, Dr. Baral. Our next speaker, Dr. Daniel de Carvalho, has redefined how we think about epigenetics. His pioneering research at the Princess Margaret Cancer Center explores how cancer cells silence key genes, and remarkably, how those discoveries can be turned into new diagnostic and therapeutic tools. Please join me in welcoming Dr. Daniel de Carvalho, speaking on fundamental and translational aspects of cancer epigenetics, from regulating retroelements to liquid biopsy applications.
All right, thank you very much for the introduction. I want to start by thanking the Foundation for this incredible honor to be here today presenting my work. So it's really exciting to share the stage with such an amazing group of scientists. So this is the title of my talk today. It's a big title, so there's a lot to unpack here. So hopefully by the end of the talk, everyone can understand what I'm talking about when I give this large title. But let's start with the cancer epigenetics. And to start to understanding cancer epigenetics, we really need to understand the human genome. So the human genome is our collection of DNA within our cells. So each cell carries about billions of letters of DNA. We have this A, T, C, G making these pairs of DNA. This is an actual printout of the human genome. We see several volumes for each chromosome, and you can imagine how big this is. The human genome contains information that allows us to build every single features in our body, every single tissue, every single cell, every single function of each cell are encoded in the human genome. So we have the recipes to build everything that we need to build within our genome. And I think, as I say this, everyone is thinking, okay, everyone knows about this. This is the most basic information about the human genome. So let's think about what not everyone knows. All of this information we're talking about to build every single cell in the human genome, every single cell in the human body, every single function in the human body encoded in the genome is only 2% of our human genome. 98% do not encode genes. They are not making these recipes to make cells. Within these, what we call non-coding regions of the genome, some people call the dark genome as a little joke around the dark matters, things that we don't fully understand. A lot of those are actually regulatory elements. So they're actually parting of the coding by actually regulating gene expression, so promoters, enhancers, and so on. But we have this half of our genome, which is called transposable elements. So these are relics during evolution, so such as when a heterovirus infects our cells. If the heterovirus copy the RNA into a DNA, insert that DNA into our cells, they become part of our cells. If this happens in a germ cell, it's actually become part of the human lineage. As the individual reproduce, the next generation will have that DNA into every single cell of their body, including their own germ cells and so on, and become part of the human lineage. So these are what we call the endogenous heterovirus. So these ERVs are 90%, you can see there. There are also other heteroelements, such as line elements, which are long repeat elements, that contains reverse transcriptase. In our cells, we have several copies, about 100 or so, functional reverse transcriptase on these line elements. So they can actually copy our RNA into DNA and allow this DNA reinserted back into our genome. So we call jumping genes or transposable elements. There's another class called signs or short repeats, especially ALU elements. So they're very small, 300 base pairs, so they do not encode reverse transcriptase, but they hijack the reverse transcriptase. So as other reverse transcriptase copy the RNA into DNA, ALUs coming together and they copy their own RNA and allow them to jump around the genome. So we call these selfish elements. The 2% of our genome is working towards the benefit of the organism. The selfish elements, they don't care about the organism, they only care about copying themselves as much as they can and reproduce themselves in this way. So if you think about what I just said, the human genome contains all the information for us to make every single cell, every single function in our body. And our human genome is mostly composed of transposable elements. Then we have two problems with this picture. One is we have the information to make every single cell, but our brain cells only make brain cells. They do not lose the differentiation, become a skin cell or a lung cell. So they need to remember the lineage as the cell divides and maintain that lineage specificity. But all the brain cells have the genes to make a lung cell. All the lung cells have the genes to make a brain cell. The other aspect is the transposable elements. If they start jumping around, our genome just become a mess. We cannot become a living organism if the transposable elements are completely unleashed and jumping around in our genome. So how do we solve these two problems? This is what we call the epigenetic. Epigenetic, we can think about this as a bookmark. A lot of tags into our genome, a lot of tags on this book of life, telling us, read me here, do not read me here. So if you have a gene that's necessary for function in a brain cell, you have a lot of bookmarks telling the cell that they can read that gene. And if you have a gene that's necessary for function in the lung cell, your brain cell has a lot of bookmark on that gene say, do not read me. So every time the cell divides, they remember this phenotype and they stay in their own lineage. The transposable elements, during evolution, we try to mutate and destroy them. But within our lifetime, what we're trying to do is silence them. So we put a lot of those do not read me tags in transposable elements. So epigenetic has these two major functions. One is this heritable bookmarking of what genes should be on, what genes should be off. And the other one is repress these selfish, these pesky transposable elements and keep them quiet and organized in our genome. So just before I move to the details of this, I want to keep in mind this is actually the central point of my lab. This is the shoulder of giants that we stand in our lab and try to develop ways to think about this, is actually understand this epigenome, understand how this code works, and understand how we can use that. So what is exactly this? I'm talking about bookmark. What are these bookmarks? So the epigenetic is this chemical modification, these chemical tags to our DNA. So one major one that we study in the lab, perhaps the one that we mostly study, is what's called DNA methylation. So we add this chemical tag straight to the DNA molecule itself in a covalent modification. And that can help the cell understand if this should be a gene that you should read or should be something that you should not read. In other words, they should be inaccessible or be accessible for the transcriptional factors. We have all the epigenetic factors, not only methylation, but we can have acetylation, we can have all the chemical modifications, not direct to the DNA, but to the histone tails, where the DNA gets wrapped in the nucleosome. So in cancer, what happens is this bookmark gets scrambled. We start to change all of these bookmarks to have advantage for the cancer cell. So what happens in cancer is you start to lose DNA methylation someplace, start to gain DNA methylation other place. Histone modifications, you lose someplace, you gain other place. So it really scrambled the whole process, creating a lot of advantage for the cancer cell. So for example, you start to have inaccessible genes become accessible. The cancer cells start to access stem cell pathways. They start to access embryonic pathways and create this huge advantage for the cancer cell. It can also shut down, it can also create genes that are inaccessible, such as tumor suppressor genes, and then they become accessible, so you can activate those genes. It's very important that this is an intrinsic feature of the cancer cell. We cannot have a cancer cell without changing the epigenome. We have profiled thousands of patients on the TCGA, the Cancer Genome Atlas. Not a single tumor tissue we can find a cancer cell that do not have this aberrant methylation or aberrant histone modification profile. The other aspect, it doesn't happen in a single place in the genome. We have these billions of letters of DNA, and this happens very broadly across the genome. We see millions of sites that we lose methylation in the cancer compared to normal. We see hundreds of thousands of sites that gain methylation in the tumor compared to normal. We see this massive change in methylation when we look across thousands of patients in the TCGA that allow us to infer the cancer type and allow us to find that it's a cancer or not. So with all of this, what our lab tries to do is take this in multiple directions. One is a very practical direction. Can you use these massive changes in epigenetics to actually profile and identify cancer? Can you use these as diagnostic tools? The other aspect we study in the lab is, okay, one function of epigenetics is heritable, gene on, gene off, and that's what cancer takes advantage of. What about the other function of epigenetics, silence transposable elements? What happened to those in cancer? And what happened to those in cancer is what's called the viral mimicry concept. So this is viral mimicry is a concept we coined in the lab exactly 10 years ago. That's the idea that if the cancer cells do need to hijack this heritable on-off bookmarking system, then they have to pay a price. The price the cancer cell pays, because they need to pay this price for do their own business, is they lose the ability to regulate transposable elements. When they lose this ability to regulate transposable elements, the cell starts to sense this as a viral infection. Our major discovery is actually when transposable elements get transcribed, the cell senses this as a viral infection, induce an antiviral response against a non-infected cell. And then as the cancer needs to transformate, they need to escape this process in order to transform. So if they sense this as a virus infection, then viral mimicry, we discover, can act as an alarm for disruption in the epigenome. It's not only about cancer, it's not only about immunotherapy, it's actually a general alarm in our genome that can tell the cell every time we start to disrupt the epigenome, something is wrong. And the way the cell finds that something is wrong is because they start to see this transposable element get waking up and induce an immune response. But they can take this to more applicable functions, such as improving immunotherapy response. If you can actually induce this antiviral response, now you can have a cell that is invisible for the immune system. The cancer cell have very little ability to activate the immune system, and now make them very, very visible for the immune system, as they have now start to look like a virus-infected cell. So what are these viral mimics? So this is a very simple concept of this pathway. The idea that these heteroelements, when they get transcribed, they start to make viral mimics. These viral mimics are double-strand RNA. We also find later on that can be RNA-DNA hybrid, as the transposable element tries to heterotranspose and reverse transcribe the RNA. We have RNA-DNA hybrid, and the cell senses this and activate the interferon response. Once the cell senses this and activates this interferon response that acts in the microenvironment, so it starts to recruit immune cells, it starts to activate a more broad response. All of this initially discovered was triggered by laws of DNA methylation. So DNA methylation is that bookmark that triggered the whole process. Later on, we find that this is much, much broader than that. There's not only DNA methylation that can trigger this, but changing histone methylation, changes in histone acetylation, mutation in chromatin remodelers, the genes that get the nucleosome and move around and open the DNA for the cell to read that piece of DNA. All of this leads to this viral mimic response. And then more recent, other groups have been showing that this pathway is even more important than just regulating chromatin stability. When you have also intronic retention, so if the cell starts to have problem in splicing, problem in processing the RNA, this intronic retention contains transposable elements, and now they are present in the cytoplasm, triggering exactly the same response. And the downstream to that is then the cell loses fitness, and then you activate adaptive immune response. Just some data showing these outputs. In the top here is when you induce DNA demethylation. The bottom is when you induce histone methylation. So for example, if you remove DNA methylation for drugs, we can induce this loss of cancer cell fitness. If you remove the ability of the cancer cell to sense double-strand RNA, to sense virus infection, you can completely rescue this phenotype. This is a paradigm change in how we think about epigenetics, not in activating tumor suppressor genes, but actually in activating antiviral response. Similar to the histone methylation, if you remove LSD1, we start to put a lot of readme tags into the DNA. They start to read the transposable elements and have the same effect. And then the output of all of that is then adaptive immune response. So a tumor that's cold, it starts to become hot, the T cells start to infiltrate. You can see this in both in the histone methylation and DNA methylation. So 10 years ago, when he published this, got a lot of attention. So in the next year, the New England Journal of Medicine published this editorial on its clinical impact of basic research, making the case that indeed, if you can use viral mimicry, we can increase visibility to the immune system of the cancer cell and perhaps improve immunotherapy response. That's the general idea I just talked about. If we remove DNA methylation, remove the tags, transposable elements wake up, the cell sense using MD5 pathway, this activates the interferon response. This interferon response talks to the microenvironment. Then the microenvironment activates the immune system. So this cancer cell becomes visible, increase antigen presentation, antigen processing. T cells infiltrate to the tumor, and then they can start to kill the cancer cell. This opened up a lot of clinical trials worldwide. Everyone's trying to find ways to combine this epigenetic therapy with immunotherapy. We have one here in Toronto. The challenge with all those trials is the DNA demethylating drugs that we have today, the current generation, that's extremely weak. So it's very difficult for us to remove enough of those tags to activate enough of those transposable elements and induce the response. But in some cases we can. And just last week, we have a trial that read out that showed amazing response in a specific cancer type. So in lymphoma patients, where I can actually remove those tags with the available drugs today. All the patients have failed immunotherapy. Once you combine these and induce the viral mimicry, you see the CD8 T cell infiltration. Now half of the patient has complete remission. 66% have objective response. So it's really transforming how this patient that initially could not respond to immunotherapy now are able to respond. And the mechanism is exactly the mechanism we described 10 years ago. But then as these trials are ongoing, in the lab, and we're helping some of those trials, but in the lab we're trying to understand better the details of how this happens. So we try to go into detailed mechanism of how these transposable elements induce viral mimicry. If you think about half of the genome is transposable element, 40% are hetero elements. If all of those can induce an antiviral response, nobody here will be alive. Everyone would have autoimmune disease. So something happens that some of those get activated, but not all of those. And we never, for 5 years, we were not able to identify everything we tried. We failed to identify those elements that activate. And then in this paper in 2020, we were able to finally identify those. What we did is we used MD5, which is the sensor for double-stranded RNA. MD5, when they sense, they bind to each other and make like this complex with the double-stranded RNA right in the middle of the complex. What that allows us is if you use this as a protection assay and we destroy every other RNA with RNases, only we can find the agonist of MD5 that we have been looking for a long time. We always think was endogenous heterovirus, but what we find is actually the sign elements or ALU elements. So if you look at the first donut, you see that total cytoplasmic RNA is mostly non-repeats, as you would expect. But then if you use the protection assay and look for the agonists of this viral mimicry pathway, we see the majority of those are signs or ALU elements. But this is at the baseline level. If you then induce those by removing the epigenetic tags, by removing the methylation, almost everything are those ALU elements. And not only every single ALU element. What we actually found here is there are usually two elements close to each other. And when they insert into the genome, they insert an invert conformation. So what that causes when transcription goes through, the RNA molecule are able to fold in itself and making this signal that the cells can recognize, make these viral mimics that the cell recognize and activate the immune response. So now that we know which one they are, we can start to study this even in a broader concept, not only in normal cells, not only in cancer cells, but across the species. And that's what we just published last month at this paper on cell genomics, showing that if you think about evolution, what happened is if you focus on the small plot first, this is high CpG regions of the genome, so these are not the ones activated this double-stranded RNA sensing. As you go millions of years back in time, as you go from left to right, or 15 million years back in time from different species, we see this no distribution. They become more and more dissimilar to each other. They decrease the signal, as you expect. But if you look at the big plot, the double-stranded RNA producing elements, the ones that actually got inserted in a way that able to activate the signal, and you go back in time, they stay exactly the same. So there's a very strong evolutive pressure in the hominidal superfamily lineage to maintain these elements that are self elements and behave as non-self. They activate this pathway. So just a function for those in benefit of the host, who is not only talking about the transposable elements as the selfish, as the pesky elements, but we actually co-opt them to activate this viral mimic and work to benefit of the host. And what are those benefits? So before I go there, this is not only the hominidal lineage. We can go and cross species and we find the same elements, aside these elements of high double-stranded RNA force that can actually activate this pathway. So the function of this is what we call the fire alarm. So we have them spread across the genome. to sense when something is wrong, to sense when you lost heterochromatin, to sense when you lost ability to splicing out introns. They act as an alarm across the genome, and every time they get activated, these cells get eliminated by this antiviral response. And this is how this fire alarm works. In every single room you go, you always see that little fire alarms. That's exactly what you see across the genome. All these red ones are elements that are able to find to be able to form this double-stranded RNA. And you spread them across the genome, and you maintain those. We do not destroy, we do not lose the ability of them to create these activation pathways. And again, as I mentioned, not only for chromatin, but also for RNA. So this is the final piece of evidence I want to show about this viral mimicry story. Is actually, this is not only important for therapeutic implication, it's not only important for species evolution, but also for cancer evolution. This is some slides that I'm showing of a pre-malignant lesion in ovarian cancer. This is a stick lesion before the ovarian cancer become a cancer, so what we call stage 0 or pre-malignancy, in a point that is very easy to treat. What you find is the first mutation you see, this p53, the most common mutation in cancer. And the first event we see is a reactivation of heteroelements. So this is a very early stage of cancer transformation. We have this alarm going on. A lot of the cells get eliminated, but some of those survive. The ones that survive, because they have this chronic stimulations, very similar to a chronic virus infection instead of an acute virus infection, what they cause is they shut down the whole immune system. So as the cancer evolves from pre-malignancy to malignancy, if you cause this viral mimicry tolerance, then you have a very cold tumor and very immunosuppressed tumor. This opens opportunities for us not only to think about therapy, but to think about interception, to think about early intervention in this cancer process. Can you stop the cancer on its tracks before it becomes a cancer? And indeed, we have some evidence for that. So this is the general model of this. The normal fallopian tube, as they become this pre-malignant lesion, we activate this fire alarm, most of the cells die. Some of them get chronic exposure and they're able to survive. All of this process can be caused by double-stranded RNA, can be caused by RNA-DNA hybrid from the transposable elements, as I just mentioned. They will cause immune suppression. If you shut down this process during the process of malignant transformation, if you use, for example, an HIV drug that blocks this RNA-DNA hybrid, blocks reverse transcriptase, we can actually prevent cancer in many models. So this is a model of 53 mutants, germline mutation. Every single of these mice, they develop cancer very fast with very high penetrance. If you treat the mice with HIV inhibitors or reverse transcriptase inhibitor, the blue line here, you delay the cancer incidence. And if you look at the bottom, the tail of this figure, it's showing that some mice, they have a normal lifespan. They do not develop cancer, even though every single cell in that mice is a p53 mutant. So that is really an opportunity for cancer interception. It's a very exciting data that I think a new direction that will go in the future. Okay, all of this is talking about the viral mimics. The fact that if you change the epigenome, you pay a price activating transposable elements. But let's forget a little bit about the biology and think about the fact that you have to change the epigenome. A cancer cell cannot be a cancer cell without a very disrupted epigenome, without methylation changing everywhere, without histone modification change everywhere. It's an intrinsic fact. The other aspect is it's broadly distributed across the genome. So it's not talking about one hotspot mutation, like one KRAS mutation, one single place in the genome, one BRAF mutation. We're talking about thousands, if not millions of places that have this mark. So if you can use this as a diagnostic tool, as a biomarker, they are extremely important. We have a broad distribution, which can give us very high sensitivity, and is intrinsically associated to cancer. It's not a surrogate mark of cancer. PSA is a surrogate mark. It's a protein mark associated to prostate tissue. If you find that in the blood, it can be prostate cancer, can be prostate tissue. With this methylation, we're talking about intrinsic aspect of the transformation that can have very high sensitivity. We published this first paper in 2018, the first proof of principle that you can use DNA methylation profile broadly across the genome and use that for multi-cancer early detection and classification. This opened up the field for this idea that was highly unaccepted in the moment, that instead of looking for mutation, or instead of looking for a single marker, you look the whole metylome, we can have very high sensitivity, very high ability to detect and discriminate. This opens multiple companies, multiple technologies competing on this field. Thousands of patients receive today this test from different approaches of profiling the metylome. And this is just an evolving field in the last few years. So we're coming close of time, so I'll go relatively fast. But the idea here is the liquid biopsy is collecting pieces of cell-free DNA that every single cell, when they die, they release this small piece in the circulation. If you collect those pieces, you can identify the presence of cancer by inferring the modification in the cell-free DNA fragment. Is this cancer-derived or is this normal-derived? I'll go to a little bit to the technology and a little bit to the clinical aspect, but before that, I want everyone to take a step back and think about the equity aspect of this. We are here in Toronto. We are in the epicenter of medical research. Every single machine that we need to have access just a few meters from us here. If you live in a northern community, if you live in a rural community, if you live in a country that is underdeveloped, you do not have access to these machineries. But it's very easy to collect a blood test and ship this sample to a lab elsewhere. So if you can actually use the blood test-derived assays for early detection, for monitor therapy response, for residual disease, you can have much broader implication in terms of accessibility for the population. So the way we're trying to do this is a technology developed called the CF-MEDIP-seq platform. In very short, is we have all these cell-free DNA fragments. We physically separate everything that's methylated in one tube, everything that's not in another. All the signal we care is in the methylome fraction, and then we sequence the methylome. Become very signal-to-noise enrichment, very data-rich, and become very cheap to sequence because now we not sequence the whole genome. We only sequence the bookmark. Instead of read all of those volumes of books, we just take the bookmark and read that page. And that's how we need to classify and identify the cancer. There's multiple clinical applications we validated over the years. I will not have time to speak all of them, so I want to focus on two very important ones. Cancer early detection and this tissue-agnostic MRD assay. So 1 minute before Janet kicked me out here, cancer early detection, we can use this to identify the different cancer types. We can identify this profile of methylation in the plasma sample and able to classify the different diseases just based on where the methylation is, what genes, what regions of the genome we get this mark. And indeed, we have the first readout of a large clinical trial running. The objective is to have much larger sample size. This is just the first few samples, 2,000 samples, across 12 cancer types. And you can see this exquisite sensitivity and specificity to detect those cancers. Highly skewed toward early stage disease, which where we want to detect. The other aspect is the MRD assay. So this is a molecular residual disease. The patient is treated with curative intent. The clinician don't know, is this patient cured or not? Should I keep treating and perhaps causing harm instead of benefit if they're already cured? Or should I switch treatment because actually the cancer is not cured yet? And what we're trying to do is identify ways to do this. So this is a clinical validation, completely blinded. The final step for us before actually this become clinical available to patients that we just published early this year show that in head and neck, we indeed can identify patients that have residual disease versus patients that do not have residual disease. Now, the clinician can change therapy, or they can actually de-escalate therapy based on results like this. And this is right now is becoming clinically available. Just as last month, some hospitals are starting to use this and revert results back to patients. So it's already have clinical impact as we speak here. So just my last slide, I thought to put some final thoughts and some acknowledgements for me to finish this presentation. The first one is the importance of basic science, right? The usefulness of this useless knowledge. People didn't think about transposable elements as something that's just a curiosity. And see what we can take of that just by following our curiosity all the way wherever the science takes us. The other aspect is teams, right? We have every single speaker talk about this. We cannot do any of this by ourselves. And I would not be here today if it was not by my lab over there that actually have taken these ideas and translate them in practice, have doing the experiments, have taken the risk of doing some of the experiments and find this exciting result. And this only happens if it's an organic collaboration. It's not a top-down that someone at a different institute try to make you collaborate. This is people with the same interests, different backgrounds, very talented people that come together to get to these results. Always be fearless, right? Don't be afraid. Take the results, whatever the results suggest you go that direction. If someone asked me 5 years ago, I would be working with evolution, I would say no. 10 years ago, if I worked with diagnostic, I would say no. And here I am today talking about this. But out of these first 3 points, I think every speaker in one way or the other, they talk about this. What I want to say is that the final point is the human side of all this process. It's not only about you be fearless, not only about you have the team, but you really need to have your support. You need to have people that inspire you, people that challenge you to be better. It can be your friends, it can be your colleagues, and for me, it's always been my family, so I'm really excited for that. Thank you.
Thank you so much. We now turn to a scientist whose genetic work in the model organism, the worm known as C. elegans, has illuminated one of life's most fundamental signaling pathways. Dr. Iva Greenwald has provided key insights into the notch signaling pathway, one of biology's most conserved systems for cell communication and cell fate decisions. Her discoveries have shaped how we understand development, tissue maintenance, and even cancer. Please welcome Dr. Iva Greenwald, who will speak on Lin12/Notch and the awesome power of C. elegans genetics.
I'd like to start by thanking, I'm sorry, I'll be like this for a minute until I get going, the Gairdner Foundation for this overwhelming honor. And honestly, having heard the talks this morning, I'm even more overwhelmed and honored and truly humbled to be part of this endeavor. So I'm going to start by giving you an overview of the novel mechanism of notch signaling. And I'm hoping my pointer, good, that's why part of the reason I'm down here. Notch is a transmembrane protein that's essentially a membrane-tethered transcription factor, where the transcription factor part is in the intracellular domain. Binding by ligand creates a pulling force that exposes a cleavage site for an atom protease that clips here and leads to ectodomain shedding. And that creates a small transmembrane stub that becomes a substrate for a multi-enzyme complex called gamma secretase that clips here and then releases the intracellular domain to go to the nucleus and activate target genes. And between Gary and me, you'll hear much more about these steps. So I have organized my talk using a slide that I showed to my undergraduates at the end of a unit on genetic approaches to biological problems, because my work has really been following this canonical flowchart, starting from the top and going to the bottom. It's been a privilege to be so close to each of the steps. And so let's talk about what the process of interest is. It's cell fate specification during animal development. And we all know we come from a single-celled zygote. And a common theme in animal development is that as the cells are proliferating, groups of cells are endowed with a certain developmental potential. We call those equivalent cells. And that means they have some choices to make about what they're going to do. So many of these choices are made when they get signals from outside the group of cells. That process is called induction, and Notch mediates those types of interactions too. Notch uniquely mediates a type of cell interaction called lateral specification. And this is when you have a group of equivalent cells and they signal amongst each other so the correct numbers and types are made. And you'll hear this generally referred to as lateral inhibition. And I'm going to show you an experiment soon that will explain why I prefer the term lateral specification. Okay, so the screen. Bob Horvitz did brilliant genetic screens to open up many different biological problems in C. elegans. And one of the screens he did was to identify viable and fertile mutations altering vulval cell lineage. And Bob actually did this himself. He pioneered the screen. And then my wonderful classmate Chip Ferguson expanded this on an enormous scale. And the screen was so effective because here's a wild type worm and it shows the vulva. A C. elegans hermaphrodite is essentially a female that can make sperm, and she can make self-progeny through internal self-fertilization. So if an animal doesn't have a vulva, she doesn't need to mate to make progeny, and it turns out she can make progeny by self-fertilization, but the eggs can't be laid. And instead, they hatch internally and eat their way out. They're only a millimeter long, so it's not as gross as it sounds. But you can see here, it's a very distinctive phenotype that you can see on a Petri dish, and we call it a bag of worms. There's another phenotype that you can see that affects vulval development. I don't know how, I can't look up. These are visible pseudovulvae, and those turn out to reflect aberrant signaling in cells with the equivalent potential to be vulval cells that normally aren't. So Chip identified a number of mutations that were dominant, mapped to a small region of a chromosome, and were either vulvaless. Well, they were all vulvaless, and some of them were also multivulva. And that's where I came in. I did much of my thesis work on something else, but I had actually joined Bob's lab to do development. And so I had my pick of genes at that point, and I chose Lin12 because I was intrigued by these different dominant mutations. And my previous work had prepared me for how I could use dominant mutations to analyze processes. So I started by characterizing the mutations in the genes they define, the nature of the morphs, and time of action, which I won't talk about. And I didn't do this alone. That's been a theme today. We hear about how we have colleagues, and I had the great good fortune to have an extraordinary scientist and a wonderful person, Paul Sternberg, work on this with me. And as we published this paper, and I'm calling it Eureka Moment 1, I'll label some of these when we felt we learned something important. And as came up earlier, I mean, sometimes you see something amazing like the cilia, but sometimes you build a lot of things by putting them, you put a lot of things together and build that Eureka moment. So students, don't worry if you don't feel that way after every experiment, but hopefully you'll feel that way sometime. So we concluded that Lin12 is a binary switch in many different cell fate decisions. And what I mean by that is if cells have the potential, say, to be A or B, and one of the fates is associated with high notch activity and one is associated with low, it's the low versus high, on versus off nature of that notch switch that specifies their fates. And we know that because I was able to identify null alleles of Lin12, and both cells became fate A. And the gain of function dominant alleles turned out to be genetic hypermorphs or elevated activity alleles. And both cells became fate B. And we found this was true in many different cell contexts, but each cell context offered a different binary choice, not A versus B, C versus D, stem cell versus differentiated cell, or I'll show you a real context in the worm later. And an important thing that was, quite wonderful timing at the time is that we knew at the time even that at least some of the decisions involved cell-cell interactions to resolve. So as we moved down our flow chart of how to do a canonical genetic approach, the next task was to clone and sequence the gene. And I did that as a postdoc. And I reported the cloning and partial sequence of Lin12 back-to-back with a paper from Spyros reporting the full-length notch sequence from Drosophila. And you can see from the title that EGF repeats was a big deal. And I should really give you some context again for the students. This was in the 1980s. We didn't know a lot. And so finding even a little bit of mammalian homology was very exciting, even if we didn't know quite what it meant. And indeed, the only developmental switch genes that behaved in this binary way, UBX and Antennapedia, that had been sequenced at that point, were Hox-containing nuclear proteins and likely transcription factors. So the presence of EGF repeats indicated at least some role involving the outside of the cell, and potentially in cell-cell interactions, which was exciting to me because of the genetics in the worm. So we actually knew more sequence than we reported at the time, thanks to a wonderful student, Kathy Weston, who got intrigued and did a lot of sequencing. And then John Yoakum not only finished and cleaned up the sequence, but cloned a second C. elegans notch gene. And we, the community was thinking of, well, how does this transmembrane protein mediate cell fate choice? And, uppermost in our mind was, how does it mediate cell-cell interactions? And so because of the binary nature of the switch and the sequence, we were particularly focusing on instructive cell-cell interactions, whether it be information that it's either a signal or a receptor. And there were other hypotheses as to the function of this membrane protein at the time that I won't go into anymore. So the next question then, if you want to know if something's a signal or a receptor, you want to know, does it function in the signaling cell or a receiving cell? And as a geneticist, our approach of choice is mosaic analysis. My brilliant student, Geraldine Seydoux, did this mosaic analysis, which was only just becoming able to be done at the time. And, and we reached two conclusions from our study for reasons I'm going to take you through in more detail than anything else that we've done. So we concluded that the simplest hypothesis to explain everything is that Lin12 is a receptor. But the point I'm going to amplify on, no pun intended, is that these cell fate specification events that involve lateral signaling involve feedback amplification of a small initial difference in notch activity. So the paradigm that we use a lot and has been so revealing for this particular point is just two gonadal cells that interact with each other and resolve which one is going to be an anchor cell and which one is going to be a ventral uterine precursor cell. Now on the right here, you can see that when an L1 larva hatches, its gonad has only two cells, Z1 and Z4, that will make the entire rest of the 155 cell gonad. And that Z1 PPP and Z4AA, which I'll sometimes call the alpha cells, collectively. These names are the lineage history of those cells, the posterior daughter of the posterior daughter of the posterior daughter of Z1, et cetera. So these cells come from different lineages, but they come together and then undergo this process to decide which one will be the AC, and there's always only one AC, it's perfect every time, or which one will be the VU. So Lin12 is and I'm using Lin12 and notch interchangeably. Lin12 is a binary switch in this decision, so 50-50 in wild type, but in a null both become ACs, so you know you need Lin12 activity in one of these alpha cells to be a VU. And the hypermorph, where you have elevated activity, high Lin12 activity causes the alpha cells both to be VUs. So Geraldine's experiment was simply to juxtapose a cell that lacked Lin12 activity to a cell that was wild type. And what she found when you look at the Lin12 null cell is it always became an anchor cell. And that says that Lin12 is acting cell autonomously in the receiving cell to specify VU fate. And that was important at the time because that pointed to a role as a receptor. But I didn't label this eureka moment because this is the eureka moment. So we were very surprised and both puzzled and energized to figure out what it meant that the wild type cell always became a VU. In an intact animal or an intact wild type cell, it has a 50-50 chance, but in this mosaic, when this cell had to be an AC because it couldn't receive a signal, this cell always became a VU. And the importance of this is it says, sorry, that the cells are communicating with each other before either commits to a fate, to coordinate their cell fate decisions. Otherwise, you might have expected this cell the wild type cell might have 50% of the time decided to be an AC, tried to inhibit the other and couldn't because it didn't have the receiving possibility. So that's why I like lateral specification more than lateral inhibition, because I like to emphasize the conversational nature of this process. So we proposed, and kind of remarkably, what we proposed has been substantiated, that initially both cells are expressing Lin12, and the ligand, which wasn't known at the time but is now known to be a gene called LAG2, and that a small stochastic initial difference that would give one cell a little bit of a head start with Lin12 would become amplified by feedback mechanisms so that the net signaler would have less receiving and the net receiver would have more receiving, and eventually they would commit to the AC or VU fates. So a couple of years later, Pascal Heitzler and Pat Simpson published an absolutely gorgeous genetic mosaic analysis in flies, a particular fly paradigm that was very good for this kind of cell-by-cell analysis, and also concluded that feedback amplification of initial differences in notch activity was important. And this was really great because when you have similar kinds of things in two very different experimental systems, even if you have multiple examples in one, but if you have it in two very different things like worms and flies, you could be more confident you're onto something fundamental. So great students, Hilary Wilkinson and Kevin Fitzgerald, who you'll be seeing a bit more later, were pushing the edge of what kinds of reporters we could do in the worm at the time. They fused beta-galactosidase coding region to regulatory sequences from Lin12 or LAG2, the ligand, and found, and just let's look at this point here, I should look at it too, that you could see that at this point, which we know by timing and anatomy is when the decision is occurring, only one of the two cells expresses Lin12 and one of the two cells expresses LAG2. And Hilary was also able to show that there was positive autoregulation of Lin12 transcription mediated by an upstream element. So it was positive autoregulation. So I just want to, and Pat and Pascal Heitzler subsequently also found evidence for a feedback mechanism that at least involved inhibition of ligand expression through a transcriptional cascade. So I just want to stop here for a moment and say that up till now, I think we can, even from these older studies, but certainly from lots of work that's been done since then, can make some general comments about the decision-making logic. The decision itself is pretty simple, notch on, notch off. But what makes it more complicated are the context-dependent mechanisms for establishing the binary choice, for creating an initial difference in notch activity between the cells, and the kinds of feedback mechanisms that are invoked. They often involve both negative and positive feedback, negative to oppose the alternative fate and positive to promote notch activity in the associated fate. So we've worked out some details of this, and I want to really highlight just an outstanding graduate student, Xantha Karp, a proud graduate of the University of Toronto, who came to my lab to do her PhD work. And she really cracked open the problem of the details by implicating a transcription factor called HLH2, which is the ortholog of Drosophila daughterless and mammalian E proteins, including E2A, which can actually substitute for HLH2. And she established that HLH2 is involved in the binary choice, and she and another outstanding student, Maria Salih, worked out involvement in the feedback mechanism I'll show you in a moment. And this group of wonderful people have more recently using some of the phenomenal tools that I only wish had been available 30 years ago, but they weren't, but they are now, and they could begin to work out how HLH2 contributes to the initial bias that gets amplified. So what we know about feedback from the work of these students is that Lin12 is positively autoregulating its own transcription. also positively autoregulating transcription of a gene called LAG1, which is the transcription factor with which Lin12 intracellular domain will interact to promote target gene expression. You obviously want them both up because you want to get those target genes going. And what this, and Catherine Lowe had implicated LAG1 here. And these three implicated the degradation of HLH2. So it's not a transcriptional cascade, but activating Lin12 directly, well, through the activation of ubiquitin ligase causes degradation of HLH2, which you need to drive the ligand LAG2 at this point. So if you don't activate, if you degrade HLH2, you don't activate LAG2, you're less likely to send a signal to this cell, so you're less likely to activate Lin12 and less likely to degrade HLH2. So that's the basis for this feedback mechanism. So feedback amplification for Notch also is involved in another cell type where it also represents crosstalk with EGF signaling. So in this paradigm, and an outstanding student, Andrew, you worked all this out, these cells have the potential to be either a type called primary or secondary, and initially both express EGF receptor and Notch, but after feedback and crosstalk, only one expresses EGF receptor and one expresses Notch. And here the feedback is a different mechanism. Activating Lin12 causes transcription of a set of genes that act as negative regulators of EGF receptor signaling, which is needed to promote the primary fate. And the positive feedback includes the autoregulation that I mentioned, but also involves the transcriptional activation of a microRNA that is needed to downregulate the GEF, FAV1, which is a negative regulator of Lin12. And that one I'll show you again one more time briefly later. Okay, so now we go to structure function studies and begin to get into mechanism. So by structure function studies, at our level of geneticists, I mean learning something about what you need for the proteins to function. And that is generally done by sequencing mutations. And at the time, many groups, including Spyros's, were generating truncated forms. And Gary and I began our first teaming up at this point with a very targeted kind of truncation that I'll describe that led us to propose the cleavage model. So, but to talk about the last kind of real solid piece of work I did with my own hands, I wanted to sequence the hypermorphic alleles of Lin12 because there are a lot of different ways you can make proteins work less well by mutation, but there aren't too many ways to give them increased activity. And when I sequenced them, and that was not like sending it out to be sequenced at the time. I mean, you can't believe how much work it used to be. But I won't go on that digression. Just don't feel too sorry for me, because at least I got a good result. So what we found was that all these mutations map to the region called Lin12 notch repeats, or between the Lin12 notch repeats in the membrane. And we now know, and Gary's going to tell you a lot more about this later, that this is the region that acts as the force sensor for unfolding the ectodomain for the clips. And we concluded, though, and I have a little quote here from the paper, that that maybe the Lin12D mutations were inactivating a negative regulatory domain. So that if you extend that, then the role of the extracellular domain is to inhibit the intracellular domain, which isn't normally what you think of receptors, right? You think of it as being there to mediate signals that activate something. And that prepared us, or it galvanized us when this very important paper came out from Jeffrey Sklar's lab. It was important for two reasons. It was the first link to cancer, and it was an important clue to mechanism. So in this paper, they showed that a subset of patients with T-cell acute lymphoblastic leukemia, which I'll call TALL, have a fusion that puts part of the Notch1 coding region of humans under the control of the T-cell receptor beta transcriptional control. And this is a figure from their paper, and this arrow shows where the break point is. So this would remove the whole EGF region, but it would leave what I just showed you seemed to be a negative regulatory region, but it would also remove the signal sequence, so you wouldn't expect this to be in the membrane. And so we reasoned if the role is to of the ectodomain is to inhibit the intracellular domain, and this truncation caused cancer by an unknown mechanism at that time, that maybe if we just express the intracellular domain, which would be the only part that would normally be in the cell, that maybe we'd see some constitutive activity. So Gary and I teamed up, and we teamed up for two reasons. One was that it wasn't clear we could even do this experiment in the worm given the state of transgenic technology at the time. And the other was, again, this idea that if you show things in parallel systems, that it has more power, especially you're dealing with invertebrates and extrapolating to vertebrates. And also, I'll be honest, the worm wasn't as accepted back then, as hard as it is to believe now. You know, people were thinking there were weird things about worms, so it also gave some power to boost the worm by, you know, associating with the fly. So I didn't label any of that eureka moment either, because this was really the eureka moment, although really it built to that. I left out something important, which is when we expressed the intracellular domain in a null background, so it didn't have endogenous Notch or endogenous Lin12, we got phenotypes associated with activation of Notch. Gary will talk a little bit more later about the flies. I'll just show you that I told you the null in C. elegans has 2 anchor cells, and when we put in the intracellular domain, you get the opposite phenotype. We'll see. Anyway, the eureka moment was that it localizes to the nucleus, and we hypothesized, so Kevin Fitzgerald, of course, was very involved in this work, and we hypothesized that the mechanism of signal transduction involves cleavage and transport of the intracellular domain to the nucleus, and may even be involved in transcriptional regulation. So Gary's going to tell you next about, if I may say, absolutely gorgeous piece of work he did that established many of these tenets of the hypothesis. And in parallel, Rafi Kopan was doing really great ex vivo analysis in cell culture, very complementary experiments that provided strong evidence for the cleavage and release hypothesis. So then the question becomes, what cleaves Notch in the transmembrane domain? Here's where we come back to the awesome power of C. elegans genetics. We did suppressor screens and found, and we was Diane Levitan, a great postdoc who was doing these screens actually looking for something else, but came across suppressor or enhancer of Lin12, number 12, in a screen for suppressors of the hypermorphic alleles. And she did the kind of genetics we do to show it was generally interacting with Notch proteins in the worm Lin12 and GLP1, and that it acts in the receiving cell. And literally, like as we were about to send off the paper, we found from work done here in Toronto from Peter St. George Hislop that familial early onset Alzheimer's disease could be associated with mutations in a gene at the time called S182 and now called presenilin-1. So that was exciting, but what did it mean? Well, what it meant for the Alzheimer's field was obvious. You know, it was an important thing for them to do, but it didn't, cell 12 null mutants didn't have canonical phenotypes. So we felt like we were on the verge of a eureka moment, but we weren't sure what that moment was going to be until a great student, Xiaojun Li, found that the reason that cell 12 null didn't have a lot of phenotypes is that there's a second functionally redundant presenilin in the worm, HOP1. And here's an example of a result. If you take away HOP1 in a cell 12 null background, you get phenotypes associated with the loss of Notch, which means wild-type worms have a rectum, and if you don't have Notch activity, you don't. That's one example. Also affected ACVU and stuff like that. Okay, so then Gary and I teamed up again. He had all these amazing tools from the study that he did to establish the mechanism. And again, Bart de Strooper, who was a fantastic scientist in the Alzheimer's field, teamed up with Rafi Kopan. And again, not knowing, we ended up with back-to-back papers that established that presenilin is required for transmembrane cleavage of Notch. Gary will tell you more about that too. So Xiaojun did something else that I think was really important at the time. Presenilin was, deduced to be a multipass membrane protein by sequence analysis, but it was typically drawn as a 7 because everybody was thinking things like G protein coupled receptors. But what Xiaojun did was he used an in vivo topology approach and identified a spartle-containing transmembrane domain. So here we were with a multi-pass membrane protein with a couple of aspartates. And the biochemists who understood this realized that this could be an aspartyl protease active site, and they proved it by getting transition state analogs that bound there. So what do Alzheimer's and Notch signaling have in common? Well, one thing is the parallel proteolytic processing events. In the case of Alzheimer's disease, an associated factor is the deposition of beta amyloid, amyloid plaques, and that comes from the cleavage of a transmembrane protein called beta APP, ectodomain shedding transmembrane cleavage. And in this case, this is the focus of much work in that field. In the case of Notch, there's ectodomain shedding, transmembrane cleavage, and signal transduction. And Gary will show you how he demonstrated that any transmembrane protein with a short ectodomain can be a presenilin substrate. One last slide about the awesome power of C. elegans genetics, which I haven't done justice to here for time. But the suppressors of the activated Lin12D hit pretty much all the core components. When you have an activated protein and you knock down things downstream, you can reduce the hyperactivity of that protein, and so we got a lot of other things. But I think presenilin was probably the star of the show from that analysis, even though the other things were important too. I also just want to point out that from looking at suppressors and enhancers, we've also identified negative regulators that have proven subsequently to be tumor suppressors and sometimes prognostic markers in TAL. One of these was called CEL10, suppressor enhancer of Lin12 number 10. And its ortholog in humans is called FBXW7. It's a ubiquitin ligase that targets the intracellular domain for destruction, thereby increasing activity, which is how it contributes to TAL. And VAV, which I mentioned as part of this crosstalk, a negative regulatory circuit, has also been implicated as an important tumor suppressor in TAL. You also get things involved in general cell biological processes because membrane proteins are trafficked, they're modified, quality control, et cetera. And sometimes they're more illuminating to those processes than the processes are to Notch. But a challenge is always to sift through and find, you know, what's integral, what's core, what's potential modulatory, because any of those things can be potential therapeutic targets. So my literally final thoughts, I keep building a crescendo and then coming back, is that why do you care? What's the basic, especially after today, like these extraordinary talks with, you know, fantastic human import, sometimes it takes a little longer to get there. Notch proteins play conserved roles and mechanisms in cell fate specification in all animals. They're continuously functioning, not just in development, but also through life in regulating stem cell homeostasis. There, as I mentioned, can be oncogenes like in TAL. They can also be tumor suppressors themselves, so everything gets flipped around. And of course, these things can be potential therapeutic targets. Gary's going to tell you later about about ways that the mechanisms and the approaches he used to deduce are being repurposed for some fantastic new tools that will be what you young people may end up doing quite a lot of work on too. Okay, do I have any time? Half a minute? Half a minute. Too many people to thank. I'm sorry, but I just need to thank first and foremost my lab. The theme today has been about teams. I've had just incredible fortune to work with people, and I made some, you know, joyful discoveries myself, but it's even more joyful when you do it with your trainees. And I want the trainees to know that, because it's just a special thing when it comes with that mentor-mentee bond. They've supported each other through the times when you don't get those eureka moments or things don't work, and celebrated with each other. And even when people didn't work on Notch, as some of these people didn't, about half, we've all been together as a team. I want to thank the worm community. The worm community is a very special place. The sharing of unpublished information and approaches and reagents is just an important part of everybody's story in our field. Of course, Gary, I don't see him. Ah, good. Got a little worried there for a moment. I owe everything to that's good in my life, and stand up for science.
Thank you, Dr. Greenwald. So he's already been partially introduced, so continuing our exploration of notch signaling, Dr. Gary Struhl has taken a complementary approach, studying how this pathway operates in living organisms to orchestrate complex developmental processes. His meticulous genetic and molecular analyses have revealed how cells make decisions in real time. Please join me in welcoming Dr. Gary Struhl, presenting Solving the Mechanism of Notch Signaling in Vivo.
I'd like to begin by thanking the Gairdner Foundation for the tremendous honor of the award for the mechanism of notch signaling. I see it not so much as an award to me, but an award for the tremendous advances in the notch field, and also to the power of genetic approaches and model systems for discovering basic principles of life, of biology. Now, I have several goals in the presentation, 3 in particular. The first is to tell you what we found. The second is to, tell you, let me go back, is to tell how we use the tremendous power of genetics and model systems to find it. And at the end, to step back and to tell you why I think what we found is important. So the mechanism is summarized here, but I'm now going to delve into it just a bit more detail. Iva's already introduced it, so this will be brief. But the process begins when the ligand contacts the receptor from the sending cell to the receiving cell and forms a bridge between the two cells. As Iva told you, and was a fundamental discovery, was that there's a special regulatory domain in the protein called the notch-negative regulatory region, which is shown here in its closed configuration. Now, the first step of transduction, of activation of the receptor, is the endocytosis of the ligand into the sending cell. And that pulls on the intracellular bridge. And that opens up the NRR and makes it accessible to cleavage. And that cleavage then triggers the next step, which is it leads to the shedding of the ectodomain. And that leaves behind only a tiny little stub of the extracellular side of the receptor. And it's that removal of the ectodomain that converts the rest of the receptor into a target for transmembrane cleavage by presenilin. And following that cleavage, the cytosolic domain goes to the nucleus and controls gene expression and thereby controls the behavior of the cells. Now, when we, our work to understand and solve the mechanism didn't follow a plan, a master plan. It actually arose step by step, starting at the end of the process, when we made one by one a series of unexpected findings, which led us up the causal chain of events until eventually we had the full narrative of the mechanism. And that's how I'm going to present the work. So for me, the story began with Iva, and especially with 2 really foundational papers in the field. The first was in 1983 when she showed that notch signaling controls the, acts like a binary switch between different cell fates. And the second discovery was her mosaic studies, which showed that notch was likely to be a receptor, and that it was a receptor for a system of feedback amplification which would let cells coordinate their choice as to what they were going to do. Now, this is a picture of Iva at the time she made those discoveries. And after these papers were published, it was, to my surprise, that her main conclusions, especially these in this paper, did not seem to be generally accepted. And so that motivated us to think if we could come up with, using the new and more powerful technologies that were becoming available, to ask whether we could actually provide evidence that would be more compelling to the field. And so that's what led to my engagement with Notch. And the experiment we did, Iva described briefly, is in order to ask whether it was receptor, and a receptor of the type in which the ectodomain keeps the cytosolic transducing domain off unless bound by ligand, which releases that repression. If that was so, we reasoned that if we just expressed the cytosolic domain, we should constitutively activate the pathway, and we should get a phenotype which is the opposite of what happens when we lose the ability to transduce. And that is, in fact, the result that we, Iva, myself, and Iva's graduate student, Kevin Fitzgerald, found was the case in both worms and flies. And I'm illustrating it here from flies. And this is the birth of the nervous system in the fly, where you get a segregation of ectodermal cells, some of which become neuroblasts, and the others choose to remain and be epidermal cells. That's the binary choice the cells have to make, and they make it in a coordinated manner normally. Now, if you don't have Notch, they all choose the neuroblast fate. But if you ectopically express or constitutively express the cytosolic domain, then they all make the opposite choice, which is to make epidermis. And I think it's fair to say that those results convinced the field that this was a ligand receptor system for determining alternate cell fates. But it was at this point we got the first surprise that led us into the mechanism, and which I was already presented, and that was that when we used an antibody that we were very fortunate to get from Spyros's lab, to look to see where was this protein, the cytosolic domain, when we expressed it. And to our surprise, it was in the nucleus. And that immediately led us to propose the hypothesis that normally, in response to ligand, the cytosolic domain gains access to the nucleus and maybe even acts in the nucleus as a transcription factor to transduce the signal. But there was an enormous problem immediately with this model, and in fact, a reason for skepticism in the field for several years, which is if you look at where the protein is normally, the native protein, even in cells such as these, which are actively transducing the signal, there's no evidence that it's ever getting to the nucleus, which meant either this model is just wrong, and this was a red herring, or alternatively, it does go there, it does do what we think it's doing, but so little of it gets there, and it is so transiently there, that you can't see it by antibody staining. It's just not sensitive enough. And that led us to ask whether, as shown here, that posed the question of, is the model right? And the model makes 3 testable predictions, and it's at this point I decided to devote my attention to testing those predictions, which I did together with the research specialists Atsuko Adachi. And those predictions are, first, that the intracellular domain should gain access to the nucleus in response to ligand. The second is that it must gain access to transduce the signal. And the third is, once it's there, it needs to be controlling transcription to control the behavior of the cell. So as you'll see in a moment, all 3 predictions were met. And this is how we did it. Okay, so to test whether it normally gets there, especially because we couldn't see it normally, we chose a strategy in which we inserted into the cytosolic domain of Notch what you could think of as a passenger transcription factor, in this case formed by the DNA binding domain of a yeast transcriptional factor called Gal4 and a viral coactivator domain from called VP16. And when we inserted that into the cytosolic domain of Notch, we wanted to know if we could see the expression of target genes that we had also introduced into the animal in response to the nuclear import of this passenger transcription factor. And as you can see here, driven through its own target gene, LAC-Z, we see beta-gal accumulation in the embryo. And so this told us that the cytosolic domain does indeed have access to the nucleus, but the important experiment here was to ask what happens in the absence of ligand, which is easy to do by just looking in a delta mutant animal. And here you can see that there's no nuclear access. So this confirmed the first prediction. It normally does have access in response to ligand. The second prediction was that it should have to get there to transduce the signal. And to test that, we took at the portion of the cytosolic domain that others had shown at that time are critical for transducing this signal. We took it out of its normal context, and we attached either a nuclear localization signal to drive it to the nucleus, or a meristalization signal which would drive it to the membrane and sequester it there so it could not get to the nucleus. And when we did that, we then expressed these two forms of the protein and asked whether you got Notch responses. And for the form that goes to the nucleus, it worked very nicely, and you got constitutive activation of Notch target genes, one of which is making the red protein here. But if you sequestered it at the membrane, there was no expression of these target genes. So that confirmed the second prediction, which is that it has to get to the nucleus to transduce. The third prediction was that when it gets to the nucleus, it's functioning as a transcription factor. And to test that, we took the following approach. We inserted into the native protein just the DNA binding domain of Gal4, or we did that together with either a coactivation domain, a strong coactivator, or with a strong co-repressor domain. And we looked to see what happened with beta-gal, and we can see that even with just the naive DNA binding domain of Gal4, you see some evidence for target gene expression. And that tells you that the cytosolic domain of Notch must have some, even if only weak, coactivator sequences in it, which is what you would want to be the case if it's controlling transcription in the nucleus. And then you can see if you add a strong activation domain to it, it's really strongly expressing beta-gal. And if you add the engrailed repressor domain, or this strong repressor domain, there's no laxi expression. So this told us these domains were doing what we think they're doing, but the important result is that this protein is effectively wild type. It transduces Notch just fine. This protein is constitutively active, and this protein cannot respond at all. So that meant that, in our view, confirmed that the protein is functioning, once it gets to the nucleus, to control transcription of target genes. And so in the end, all three predictions were met. And therefore, we view the model as these results show that the model is basically correct. And I should add that there were several other labs which made very significant contributions along the same lines around the same time. But it was in the process of pursuing these experiments that we stumbled on the second unexpected result. And that's shown here, which is that it seemed as if the cleavage event, which would release the cytosolic domain to go to the nucleus, that cleavage event seemed to be happening within the plane of the membrane, in the transmembrane domain. At the time, that was an unprecedented, virtually unprecedented finding. Now, the result that suggested that were a couple different results, but from our work, the result that suggested it is when we first set up this system with the Gal4 technique to ask if the cytosolic domain got to the nucleus, we didn't know where, if a cleavage occurred, where it would occur. So we put it in many different places. And somewhat to our surprise, we found that even if we moved the Gal4 VP16 domain right up against the transmembrane domain of Notch, it still got to the nucleus in response to ligand. In fact, it worked very nicely, which meant that the cleavage would have to be upstream. Around the same time, this dovetailed very nicely with Iva's work, in which she had identified cell 12, now we know worm presenilin, as necessary for Notch transduction and signaling in worm. And at the same time, Bart de Strooper had found that presenilin in vertebrate systems seemed to be required for the transmembrane cleavage of beta amyloid precursor protein. And so it was at that point that we wanted to ask the obvious question of whether presenilin was responsible for the nuclear access of Notch in response to ligand. And so Iva and I decided to collaborate again to ask whether that was the case. And we used the Gal4 technique, which we had been using to work out the mechanism to do that experiment. And this is the result we obtained. The experiment we did was basically first using the native protein to show, which we already knew, that it's expressed in the embryo. It gets to the nucleus in the embryo. But in a presenilin mutant, it doesn't. But more importantly, we used a severely truncated form of Notch, which has just a tiny bit of the ectodomain left and the transmembrane domain. And that too normally gets to the nucleus, but does not get there in the absence of presenilin. But if we then use a form which is purely cytosolic, it doesn't care whether presenilin is there or not. So these experiments tell you that presenilin is critically required for ligand-dependent nuclear access of Notch. So, and this was similarly supporting findings in mammalian cells, came also from others at around the same time. So in the process of doing this, we stumbled on one more surprising result. And that result, we found that result because we wanted to understand what ligand contacting the receptor would do to induce the presenilin cleavage. And presenilin normally doesn't induce Notch cleavage in the absence of ligand. So something had to tell it when to cleave in response to ligand. And that result, which we stumbled on, was the surprising finding that the only thing the presenilin seemed to care about was the size of the extracellular domain. And that the implication of that is that the key event to getting to this next step was the removal of the ectodomain of Notch to make the ectodomain small. And that converts the receptor into a substrate or target for presenilin cleavage. Now, the results that we got to test this is shown on the next slide. And here I again work with Atsuko Adachi. And we again used the GAL4 technique to ask about nuclear access. And what we wanted to know was, is there some special attribute of Notch, something that presenilin sees to help it target Notch for cleavage? And so we began simply by first removing the cytosolic domain. And then, in addition, we removed the extracellular domain and replaced it with a single MIC tag. And so now all that's left is just the transmembrane domain of Notch. But those deletions just made it a better target for presenilin. So then we thought, well, maybe it's the transmembrane domain. So we replaced the transmembrane domain with the transmembrane domain of multiple other unrelated proteins, and then again found that it didn't matter. Presenilin would cleave them all because when you remove presenilin, none of them got cleaved. And so this led us to what was at the time a seemingly bizarre possibility, which is that presenilin only cared about the size. And to test that, we essentially repeated these experiments, but in this case, we started with a single MIC tag, and we just added MIC tags one by one to see if that changed the efficiency with which the cytosolic domain got to the nucleus, and as you see here, it did. So what this told us is that the only thing presenilin cares about is the size of the ectodomain, and that led us to the conclusion that the important event here is the extracellular cleavage and shedding of the ectodomain, and that's what triggers the rest of the mechanism. And I should add here that others also contributed to this, including Pan and Rubin, who found the protease responsible for the ectodomain cleavage. Now, it was at this point in the process of doing the experiments I just described that we stumbled on yet one more unexpected observation, and that is shown here, which is that in order to trigger that cleavage, the ligand had to be endocytosed into the sending cell. And this also dovetailed with results from the lab of Mark Muscovich, who similarly found a requirement for ligand endocytosis. And what I'm now going to describe is that we wanted to know why you needed to endocytose the ligand. And I'm going to describe experiments in which we found that the explanation is that by endocytosing ligand into the sending cell, we are creating mechanical strain on the ligand receptor bridge, which is what is opening up the NRR, the regulatory domain, for cleavage. So for us, the experiments to do this began in earnest when I began to work with a graduate student, Weidong Wang, and we identified two proteins which are critically required in the sending cell for Notch signaling that were specifically required for the targeting delta into the clathrin-mediated endocytic pathway. The first of those proteins, actually been identified by others, but is a E3 ubiquitin ligase, which selectively ubiquitinates the cytosolic domains of Notch ligands. And the second protein is a generic endocytic adapter protein called epsin, which if it sees any cargo protein that's ubiquitinated, it targets it for endocytosis. Now, the important point was that both of those were critically required for endocytosis of the ligand and for signaling, which all sounds fine, but if you actually think about it for a few moments, you realize it creates a conundrum, which is if you endocytose the ligand into the sending cell, it's no longer available to see the receptor. So why do you need it to activate the receptor? It made no sense. And the explanations we came up at the time were the two general classes of explanations. One we call before models, and the other we call after models. And before models are, you could think of them as recycling models. And the idea is that when delta first comes to the surface, it's in a protoform that is either not able to activate the receptor, or which maybe is put on a portion of the sending cell surface where it doesn't have access to the receptor. And it needs to undergo at least one round of endocytic recycling to be modified or to be moved to a place where it can productively engage receptor. And so that was one class of models. The other class of models we called after models. In those models, you actually don't need to endocytose the ligand until after, as the name implies, it conjuncts receptor. And then when you try to endocytose it, those are the ones in which you put strain on the regulatory domain, which I'm here depicting as a spring, and that's what opens it up to cleavage. So we then wanted to know which of these two general classes of hypotheses are correct. And it should be clear that it's absolutely critical to resolve this to understand the basic mechanism. And it took us about 10 years. I was fortunate to work with a really great postdoctoral fellow, Paul Langridge, who had enormous patience and willingness to devote himself to this task. And we'll see, the way we did this was by first performing in vivo experiments in which we essentially invalidated those two classes of recycling models. And then we were able to perform experiments where we got positive evidence for pulling models. And that's how we resolved the mechanism. So the first set of experiments, this is an example, a key example, the experiments we did, which helped refute the before models, is that we replaced the ectodomains of delta and Notch with the ectodomains of completely unrelated ligand receptor pairs. And in this case, we're showing mammalian follicle stimulating hormone. It's a secreted mammalian hormone. There's absolutely no expectation that would have to be recycled to be activated. And it's the ectodomain of its cognate receptor to create these chimeric ligand receptor pairs. And we reasoned that if you don't need to recycle this to activate it, there's no reason why you would need endocytosis. You should bypass the requirement for endocytosis. And when we did the experiment, that is not what we found. And just to show you what we found, where we assayed for this was in the developing wing. And normally, the dorsal half of the wing and the ventral half of the wing signal to each other through Notch to induce target gene expression and behavioral changes, cellular behavioral changes, along the interface between the two halves of the wing. And that turns on, for example, this yellow protein, which is from a Notch target gene. And what you're looking at here is large clones of cells which ectopically express this chimeric ligand. And the surrounding black cells are expressing the chimeric receptor. And wherever the two interface at the clone border, you see ectopic induction of the Notch target gene. So we've reconstituted signaling, but the important result is that if we block entry of this ligand into the endocytic pathway, you get no ectopic activation of Notch pathway. And so that's telling you that that's arguing strongly against recycling models, because otherwise they should bypass it. Now, in the process of doing those experiments, we got a huge bonus result. And that's when we looked carefully at what's actually happening to the ligand and the receptor when we're getting signaling blocked. And that is what we found was something quite remarkable, which is if the ligand cannot be endocytosed, the receiving cell actually eats a portion of the membrane of the sending cell, including the entire ligand. And this you can see here, this is the clone. and in this case, the FSH delta is red. And you see the neighboring cells, which have puncta, which have the receptor, actually have incorporated the ligand into some of those puncta, especially right next to the clone. Now that was important because it told us that not only do you not need it to recycle to modify the ligand, you also don't need it to move it to a part of the cell where it can see ligand, because for the receiving cell to eat the ligand, it has to see it. So this essentially invalidated both of the main versions of recycling models, which left us with pulling models. And here the challenge was the after models. And the challenge here was, how can we show that you actually need mechanical force in vivo? That's the operative agent in vivo to activate the receptor. And that proved to be a challenge. But the way we resolved that is we did the following experiment, which was kind of remarkable that it worked, actually, which is we reasoned that if this domain is functioning as a force sensor, maybe we could replace it with a truly bona fide force sensor and reconstitute delta notch signaling that way. And we chose as our heterologous force sensor the A2 domain of von Willebrand's factor, which is a blood clotting factor. And this domain is cleaved in response to mechanical stress, in turbulence in blood at injury sites. And because it's involved in a large number of genetic blood clotting diseases, it's been the subject of intensive study by biophysicists for many years. And it has been shown to function as a bona fide force sensor. And even more importantly, because it's associated with blood clotting diseases, those diseases are genetic mutations in the A2 domain, which change the tuning, the threshold, the force threshold necessary to open up the domain to cleavage. And so we could then choose different forms of this domain and ask whether any would have the right tuning to reconstitute notch and signaling. And I want to also emphasize, as you can look at the structures, they're completely different, and as is the cleavage site. But both are actually cleaved by the same family of metalloproteases called atom proteases. So that's the experiment, and the result was, in fact, we could reconstitute delta notch signaling, but only if we had the right tuning. So this is the native native A2 domain, and this is the same assay, and you see induction of the target gene, but only along the normal interface between the dorsal and ventral halves of the wing. And this is even though you had clones which were confronting ligand to receptor. But if we then look with a disease-associated form of the A2 domain, which has a lower force threshold, now we could see at the edge of the clones ectopic induction of the notch response. So we could reconstitute it, but we do not see that when we block the ability of the ligand to enter the endocytic pathway. So with that result, that confirms basically the pulling aspect of the model. And I wish to also note that other labs contributed very significantly to evidence that pulling is actually the operative mechanism in vivo. So now we have essentially, starting from the back end, worked our way up one by one through the causal chain of events, which constitute a narrative, a complete narrative, effectively, for the basic mechanism of signaling. So I would now like to step back and ask, why is this important? And there are several points I want to make. The first is the way we did these experiments, the approach we took, shows the tremendous power of genetic approaches in model organisms to not only tell us about the logic of how our pathway works, but actually to get to the molecular mechanism. And it could be that it's in the nature of the notch signaling mechanism that it was particularly tractable to the kinds of experiments we love to do. But even so, it's a testimony to the power of the approach that we were able to get as far as we got. The second point I want to make is the value of the early studies in these systems, and especially when it became available, the ability to create transgenes to express altered forms of notch which have either constitutive gain or loss of activity in any cell you want, in any tissue you want, and now in any biological system you want, to actually determine and to understand what Notch is doing in virtually any context. And that, together with, as Iva mentioned, the early evidence that Notch was implicated in disease. And virtually any gene involved in controlling patterning and growth, we now know they're virtually all involved in things like cancer, because that's what cancer is. But what's important is, up until that point, when we had both the foundational studies and the link to disease, at that point there may have been 10 to 20 papers a year, but shortly after that, there's this enormous explosion of publications to now about 2,000 a year, which shows how these early studies to get at the basic mechanism fostered an explosion of work and discovery in Notch and its relevance across tissues, disease, everything, basically. The third point I want to make is, by getting at the basic mechanism, we've uncovered what seem to be general paradigms which are crucial for cell-cell interactions and signaling. Iva talked about the connection between Notch signaling and beta-APP signaling, and now as of now, those were the original 2 examples, but there are now over 150 examples where pre-signaling and the cleavage and these sorts of mechanisms are now known to be occurring and to be biologically relevant. I want to tell you another example, which is very recent, which is in some ways, well, it is very remarkable, which is in the last couple of years, we were able to use our methodologies to do a genetic screen, effectively a genetic screen, to ask what are the kinds of domains that could serve as force sensors to reconstitute delta Notch signaling. And we queried about 45 to 50 domains, some chosen at random. Some were chosen because they were from other proteins that mediated contact-dependent intracellular signaling. And the remarkable finding is we got about a quarter of the domains we assayed worked. But as you see here from AlphaFold predictions, they encode very different types of structures. And not only do they work and can substitute for the Notch regulatory domain, they all depend on cleavage by the same protease that cleaves the Notch negative regulatory region and the A2 domains. So this is really, it's so new result, I still haven't fully absorbed what it means, but it's telling us something very important about the mechanism that targets the regulatory domain for cleavage and how force sensing works. And the final point I want to make is that our use of chimeric proteins, what we had to do to interrogate the mechanism, also led us to reveal that this system is remarkably modulatory. And in principle, the mechanism doesn't care about any special attribute of either the ligand or the receptor. All you really need are extracellular domains which will bind to each other. You need a force sensor, and you need a transducer, typically a transcription factor. If you have that, you have the basis for a delta Notch-like signaling mechanism. And in fact, so that's been repurposed in what is now a rapidly expanding field called synthetic Notch signaling, where you can essentially engineer designer signaling systems to work in vivo. And this is already proven to be very valuable in cancer treatments. It's being rapidly advanced in terms of tissue engineering. So basically, from knowing the basic mechanism, we've been able to go to a relevant treatments for disease and for human health. And on that, I'd like to conclude by acknowledging many of the people I've talked about. And I most especially want to acknowledge Iva. I would not have worked on Notch without her. It's been a journey of about 30 years. And it's been really tremendously engrossing and rewarding. And I wouldn't have done it without her. So I'm very grateful to her. I want to also emphasize the people we work with, Kevin Fitzgerald from Iva's lab, Atsuko Adachi, Weidong Wang, and Paul Langridge, also my support. And I also want to emphasize, as I said in the beginning, I don't see this as an award to me. I see it as an award to the field. And there have been many people here, some I've mentioned or put on the thing. There are many others. So with that, I think I'll conclude. Thank you.
Our next early career investigator, Dr. Nika Shakiba from the University of British Columbia, explores how stem cells compete, cooperate, and evolve within developing tissues. Her work blends bioengineering and developmental biology to uncover the rules of how cells decide who thrives and who dies, insights that could transform regenerative medicine. Please welcome Dr. Nika Shakiba speaking on the molecular rules of embryonic stem cell competition.
Well, first of all, I just want to say it's a great honor to be speaking on the Gairdner stage, and I really want to thank the Gairdner laureates, Drs. Greenwald, Struhl, and Artavanis-Tsakonas, who chose me to be the ECI speaker in their session. I'm an engineer by training, and although my lab is a stem cell engineering lab, and we're motivated by the idea that you can turn stem cells into engineered entities for regenerative medicine, our core belief is that in order to do that, we have to spend our time to first uncover the molecular rules that determine how the stem cells behave. And today I'm going to tell you a story in that vein, understanding the molecular rules of competition between our cells. And this is a story that's led by a very talented PhD student, one of my first two students, Kieran. So if you think back to your origins, perhaps you can't remember that far back, but we all started from a single cell, the fertilized egg. And this cell has the remarkable job to go through a series of very well-defined divisions to create first this cluster of cells, this blastocyst, that then has to coordinate together. This group of cells, this society, has to coordinate to make our tissues and organs and ultimately build our bodies. And the way that this is done, well, these cells have to communicate with each other, and we heard a lot about notch signaling. So these kinds of fundamental mechanisms that cells use to communicate with each other are required for that coordination. But what's interesting to my lab is that sometimes it's not a coordinated effort. Sometimes there's riots. And there's competition between the cells as they're building our bodies. And this is a relatively newly discovered cellular phenomenon. It was first reported in 1975, this idea of cell competition in the Drosophila. And what these studies did is they derived these minute mutants. So these are ribosomal protein heterozygous cells that can fully form a fruit fly. So in the embryonic development, they have no trouble creating a viable fruit fly. The fruit flies are largely normal. They have slightly shorter bristles, as you'll see, but they're functional. But when you mix these minute mutants with wild-type cells, normal cells, and you allow that fruit fly to grow, what you'll find is that the winner cells, the wild-type cells, will actively eliminate the minute mutants from the embryo. They seem to do this in a context-dependent way, so the losers only get eliminated if winners are nearby. And it seems to be that this is contact-mediated or proximity-mediated, so the winner needs to be near the loser to do that. Now we know that through more recent examples, this is a conserved mechanism. So for example, when the zebrafish is being developed, cells that are in the tissue patterning process and receiving slightly higher or lower levels of Wnt signaling will get eliminated by their neighbors in order to make sure that our tissues pattern correctly. And now we know most recent examples in humans. So if you take embryonic stem cells from the embryo of humans and you grow them in vitro, you'll find that certain genetic variants, which are those in green, can actually actively eliminate loser cells, the wild-type normal cells from culture. And this is an active, again, contact-mediated killing mechanism. In one of my favorite examples, a lab, Jinwoo's lab in the US, actually did this competition arena where they took embryonic stem cells of different species and they decided to compete them against one another in vitro. And what they found was that, again, this is a conserved mechanism that even across species you can have competition. And they showed that, for example, if you take mouse or rat cells and compete them against human or rhesus, they'll eliminate human or rhesus. And unfortunately, the cow is the ultimate loser. So the best example that's understood in cell competition in the context of mammalian systems is in the mouse. And we know from looking at mouse embryonic development that competition between cells is conserved across developmental time points. So in that ball of cells I mentioned, the blastocysts, the cells that are taking slightly too long to make their early life decisions, what kind of cell to become, primitive endoderm or epiblast, those cells get actively eliminated by their neighbors or that are saying, hey, you're taking too long, the embryo needs to develop, we need to get rid of you in order to progress. And then after that embryo implants into the uterine wall and starts to develop into the body, at that stage of development, about a third of the cells inside the embryo that are responsible for making the mouse get eliminated. A third of the cells. That's pretty remarkable. And we now know that we can actually engineer those winners and losers. So for example, a study has shown that you could perturb the levels of MYC, which you might recognize as an oncoprotein. And if you have slightly higher levels of MYC, that will make you a winner and you will completely take over the mouse at that stage. Later on, as our tissues are forming, for example, as the skin is developing, you see, again, examples of cell competition that help to make sure the skin's architecture develops correctly. What's good and handy for my lab is that actually you can take, as I mentioned, those embryonic stem cells out from that early stage of embryonic development, put them in the dish, and these so-called mouse embryonic stem cells retain this competitive phenotype. And we now have this kind of hallmark assay of cell competition where you can actually take genetically different mouse embryonic stem cell lines, so they're on the same genetic background, but they have perturbations to key genes involved in cell competition, and you can paint them with fluorescent proteins, different colors, and then the competition phenotype is seen when you compare what happens when these competitors are grown on their own, where you'll see that they grow normally, they're quite viable, but only when you mix them in co-culture, you see that losers are eliminated. So again, it's this context-dependent death that defines competition. Now, using these in vitro and in vivo systems, we've learned some things from mouse embryonic stem cell competition. Well, first of all, we know that there's certain genes, some oncogenes you might recognize, like P53 and MYC, that if you perturb, you can make a winner or a loser. So you can make winners by knocking out P53, you can make winners by overexpressing MYC. And mTOR activity seems to also be important. So higher mTOR makes you a winner. But all of these seem to be sufficient, not necessarily necessary, I should say, for competition. And this led my lab to ask, well, what is the fundamental necessary molecular node to make mouse embryonic stem cell competition happen? Now, it turns out we don't have to fly blind. Because this is conserved, we can turn to really robust data from the fruit fly literature that has shown us a lot about the role of the ribosome in the context of competition there. As I showed you, minute mutants have ribosomal protein perturbations, right? So you lose a copy of a ribosomal protein and you become a loser. And since then, studies have shown that really any or many ribosomal proteins, if you lose a copy, you're a loser. But perhaps what's more interesting is that what's necessary for a loser to die in competition in fruit fly is the ribosome. So losers, as they're dying, they will activate XRP1, which is a key transcription factor that then triggers the stress response in these cells, the integrated stress response, and this ultimately shuts down translation. It shuts down the ribosome through EIF2 alpha phosphorylation. And so looking at the Drosophila literature, we hypothesized, well, maybe the ribosome is also the key node, the missing node that we haven't been looking at yet in mouse embryonic stem cell competition. And so Kieran turned to our collaborator, Tristan Rodriguez, who generously shared with us mouse embryonic stem cell lines that are known winners and losers. And so we took a few of these lines. So for example, we took wild type cells and BMP receptor 1A knockout cells. BMP receptor 1A knockout cells are actually losers when you mix them with wild type. And P53, as I mentioned, if you knock that out, that's a super competitor. That can actually eliminate the wild type cells. So we had these two competition assays where in one case, wild type cells are losers, and in another, losers, and in the other, they are winners. And we recapitulated these cell competition assays as we expected to see. We showed that when you grow the cells on their own, they're quite viable, they're pretty normal. But then in co-culture, you start to see that the winner overtakes in both situations. And this is perhaps more clearly shown when you look at the ratios of the cells on the right. Now, Kieran said, well, I think the ribosome is involved, so we can't do transcriptomic analysis. And we decided instead to take the cells at this day 3 time point where you start to see competition emerging, and we did proteomic analysis. So we sorted the winners and losers from the co-cultures and the monocultures, and we did mass spectrometry. And we looked to see what's changing. do we see ribosomal terms? And when we did GSEA analysis on this omics data set, the first thing we noticed is that even in the absence of competition, so before the competitors have even had a chance to compete, it looked like our losers maybe have some level of deficiency in ribosomal biogenesis or assembly. For example, you see that the large ribosomal subunit seems to be dampened in our losers. And then in co-culture, when they're actually competing, you see the losers, the really bright blue here, indicates that they're shutting down ribosomal assembly and biogenesis processes. On the other hand, winners seem to elevate those processes. And so now we asked, well, like Drosophila, is the integrated stress response involved? And so we turned to our collaborator, Ramon Klein-Geltic, who did these Western blots for us, and we actually don't see phosphorylated EIF2 alpha, so we don't think the integrated stress response is involved in mouse embryonic stem cell competition, and our omics analysis actually corroborates this result as well. So not ISR, where is the ribosomal bottleneck then in mouse embryonic stem cells as they're competing? For this, we then turned to Li Vu, speaking of team science, a lot of scientists involved in supporting our work. We turned to Li Vu and we did polysome profiling. And what this allowed us to see is to actually measure the relative quantities of the different parts of the ribosome. So the monosomes, the 40S, the small and large subunits, as well as the fully formed ribosome, the monosomes, as well as the polysomes. So these are multiple ribosomes bound to the same piece of RNA and translating it. And the signal that came out of these results was that we always found that the losers seem to have a relative deficiency in the large subunit, which is something that also came out from our proteomics analysis. And this persists during competition. Does this result in ribosome heterogeneity? We see winners and losers have different relative levels of ribosomal proteins. I didn't show you the heat maps for these, but they have different levels of individual ribosomal proteins. And maybe there could be different pools of ribosomes that are heterogeneous from one another. And around this time, Maria Barna's lab had shown that actually mouse embryonic stem cells have heterogeneous ribosome pools that seem to translate different pools of transcripts. And we were seeing the same differences in key ribosomal proteins that her lab had shown to be involved in heterogeneity in our data set as well. So we thought, well, maybe that's involved in mouse embryonic stem cell competition. But when we actually pulled out the fractions, the free subunit, monosome, polysome fractions of our polysomes profiling data, and we did mass spec on that, we looked at the ribosomal protein stoichiometry, and we didn't see any difference between competitors before or during competition. So this made us rule out ribosome heterogeneity. That's not involved here either. Okay, great. So does this mean anything? Are the losers actually shutting down translation the same way that happens in Drosophila as they're losing? And so we did an experiment using SILAC labeling. So this is where you basically add amino acids that are heavy labeled into the media. So we did this about 6 hours before we assayed the cells, and then we did mass spectrometry again. And what we found was that losers, before they're competing, so in the absence of competition, they're translating at the same rate. So they're producing nascent peptides or proteins at the same rate as the winner cells. But it's only when they compete you start to see that they're shutting down translation. And both wild-type cells and BMP cells do this when they're losing. And what was interesting to us is that when you actually look at do omics analysis, GSEA analysis on the nascent peptides that the winners and losers are producing, what you see is that actually losers are producing target genes of the MYC pathway, for example, which is a key fitness-associated gene in cell competition. They're expressing actually higher levels of MYC target genes than winners before they're competing. And it's after competition that they start shutting down these fitness-associated genes. So we think that there's some kind of positive feedback loop where losers are then enforcing down regulation of the expression of fitness genes as a result of their ribosome bottlenecks. And so this is now our updated working hypothesis. We've observed that it looks like losers have bottlenecks in their ribosome production. They have fewer ribosomes when they're competing, this limited ribosomal pool, and they start to shut down translation during competition. And so we've now amended our hypothesis to say that because of these ribosomal bottlenecks, this is leading losers to exclude a subset of their transcripts. They're not translating a subset of transcripts. And as a result, this is making them lose. This is the molecular mechanism we think that's involved. And now we're working on assessing this using a synthetic biology technology out of Jean Yeo's lab called RiboStamp. And the idea of this technology is they took a ribosomal protein, in this case RPS2, and they fused it to a base editor, APOBEC. And APOBEC basically allows, as that ribosome is assembled and it's translating a transcript, it causes mutations to occur in that mRNA. And so when you do next generation sequencing, you can actually read out not just the abundance of a transcript, but also how much that transcript is being translated, or so-called the translatome. And so we've generated these genetically engineered mouse embryonic stem cell competition lines. With this RiboStamp, we've confirmed that RiboStamp's expressed. The cells look like, at least transcriptionally, they're normal. They still compete the normal way. And when we look at RiboStamp editing, we see that they're successfully editing above the baseline levels in the coding sequence in the 3' UTR, which is where you would expect to see it. And we've now just recently, hot off the press, done this single-cell RNA-seq time course run where we've allowed our competitors to compete with each other and assay them every 12 hours in competition. As I speak, Kieran is working on processing this data and looking for the edits to the transcripts to see how the translatome is changing in competition, so stay tuned for those results. I just want to loop back to the kind of central vision of my lab, which is not sole vision of my lab, but we're really wanting to understand how interactions, competition, cooperation between embryonic stem cells and pluripotent stem cells shapes how our bodies are built, and we fundamentally believe that we have to unlock these rules in order to port them over to stem cell engineering to make cell therapy manufacturing processes more robust. And the best way to do this, we think, is through team science. So in a recent international consortium that I co-founded with Drs. Maria Abushakra and Nozomu Yashi, actually Maria's in the audience with her daughter there, we created this international consortium around the central vision that you could bring computational biologists, mathematical biologists, empirical scientists in developmental biology and stem cell research around the central vision of distilling these rules of development together and making a virtual simulator of human embryonic development that will help us do that. And by doing that, then we will really understand how our bodies are built. Then we can really engineer cells for therapeutic purposes. And I just want to end by acknowledging the people behind the science. My team has quite grown in the last few years, and I'm really privileged to work with these really bright minds that make my day-to-day life fun. And I really want to thank the funders who enabled us to continue tinkering in the lab over the years. Thanks so much for your attention.
Thank you, Dr. Shakiba. To close this extraordinary program, we turn to a scientist whose work has been central to our understanding of the Notch pathway. Dr. Spyros Artavanis-Tsakonas' research has shed light on how the signaling system drives cell differentiation across species, inspiring decades of continued exploration. His career exemplifies the power of curiosity and persistence in science. Please welcome Dr. Spyros Artavanis-Tsakonas, who will present Notch signaling pathway, a paradigm of biological complexity.
I think it's on now. All right, okay. Well, I'll say again, thank you very, very much. It's a tremendous honor to be recognized in this manner. But I should also say that, how do I?
That's your pointer?
My pointer.
Advancing yourself here?
Yeah, well.
Go ahead.
Here we go. So I should say, however, that everything I say today, every single thing I say today, it's done essentially by other people. If it was not for my postdocs and my graduate students, I certainly would not be here. I will not acknowledge them in detail, simply because there are too many to acknowledge, but I can assure you that they got the prize, not me, okay? So, notch, you've heard now, you're all experts in notch, mechanistically and otherwise, thanks to Iva and Gary. But I'll give you a little bit of a different perspective, or try to put things into perspective, but more general biological perspective, for better or for worse. So notch is a very old gene in the sense that it was uncovered. I mean, it's millions of years old, but it was uncovered by the magical group of Morgan and his associates, and more than 100 years ago. And the attribute, the essential part, was that they found that the mutation, Dexter found that the mutation is an X-linked mutation, and in a heterozygous form, it has this notch, the wings that are notched in the margin, hence notch. How many times I was asked why it's called notch, I can't tell you, so you forgive me if this is an obvious thing. And also, what at the time was recognized that loss of notch function, completely loss of notch, is lethal, and we'll come back to that. I'm asked very, very often, why did I start more than 35 years ago working on notch? And I was finishing my PhD in Cambridge, and I was thinking of doing molecular developmental genetics because the technology was becoming available to do that. And, and there was one review on developmental genetics by Ted Wright, who was saying this phrase, that if there was one gene to look in development, it should be notch. That essentially hooked me. I mean, this was like the hook. I still have the hook, mind you, so I'll tell you a little bit about the prehistory of notch. I mean, you heard a little bit about the history, a little bit. But I'll tell you a bit of the prehistory. So after this discovery of the mutation, nothing much was happening in terms of notch biology until Don Poulsen, an extraordinary embryologist, decided to start I was analyzing X chromosome deletions and relating those to phenotypes. At the time, this was extremely important because people did not believe that genes had anything to do with morphogenesis, nothing, and embryonic development. So Don, who was doing his PhD under these two Drosophila giants, Dobzhansky and Sturdivant, had been correlating. This is a drawing by Don that's hanging in my office, who gave it to me. What he found, he essentially established by doing analysis of deletions, that notch maps at 3C7, which is a chromosomal location in giant chromosomes. But he also looked at the phenotype of the null mutations, and he saw that if this is now a developing embryo stained for neural structures, you can see that the neural structure is fairly orderly, if you will. And if you don't have notch, everything, there's a hypertrophy of the nervous system, but the cellular phenotype was that the nervous system, the neural structures were at the expense of epidermal structures. So precursor cells that were supposed to give rise to both either dermoblasts or neuroblasts, in the absence of notch, they were giving rise to nervous system. To solve the problem, of course, to start looking at the molecular genetics, with the emphasis of molecular. In this, you needed to clone we had to clone the gene. And at the time, again, I was working in Dave Hogness's lab, and Mark Muskevich, with whom I'm still a friend, and we're still working together, actually. His picture above is then, and the picture below is now, just so you can recognize him. We started isolating overlapping clones. He was working on a protein and a gene that was in 3C10. So we could now start isolating overlapping clones in the gene libraries, phage libraries that actually were constructed in Dave Hogness's lab, to look at overlapping clones to walk to 3C7. However, the problem was that we stuck in a repetitive sequence, and we just could not advance. To the rescue, the only lab at the time, working on notch, and that was Bill Welshon's, an extraordinary cytogeneticist who spent his life looking at 3C7 and doing cytogenetics around this locus. And he had defined, he had uncovered or actually generated, in fact, a rearrangement, a chromosomal rearrangement that allowed us to jump over this repetitive sequence. And that, of course, allowed us to clone the gene. Oh, sorry, I did. To clone the gene, and in many ways established the field, the beginning of the field, because without having these molecular tools, we would not be doing that. We would not be here today anyway, all right? Now, I want to make a point, because I'll come back to that. I told you about the nervous system, the neural development abnormalities, but something which is extremely important to mention is that notch activity is pleiotropic. It is incredibly pleiotropic. It's a pain in the neck pleiotropic. It is involved in everything, almost, okay? So that is something that I want you to keep in mind. And it's involved in proliferation, it's involved in differentiation, in different cell fates, I mean, as we heard earlier today, and so on. It's involved in apoptosis, controlling of, and movement. So all the necessary developmental processes that can make a three-dimensional organism, if you will, all right? Nevertheless, in spite of this pleiotropy, there are a few things that are constant, let's say, or fundamental in the logic of how notch works. You heard about at least one of them, which is this notion that this membrane, these interactions via notch allows a cell to tell the neighbors what to become. Whether I become a red cell and you don't become a red cell, or you become a white cell, depending on the context and so on. So this is, of course, absolutely fundamental in morphogenesis, because lineage segregation is almost the quintessence of morphogenesis, almost. And indeed, as we heard earlier, stem cell systems are all affected by notch. Any stem cell system that we know of can be influenced by tweaking notch activity. How this is done, we heard all the details about it. I'm not going to tell you more about it. It's done with a membrane-bound receptor and a membrane-bound ligand, with all the delicious intricacies that Gary and Iva outlined, all right? And their talk actually pointed already to some aspects of complexity. I mean, by extrapolating on what they said, all right? And I'll come back to that, of course. Now, one thing I also want you to remember, that abnormal signaling, so if you modulate notch in an abnormal way, this is either abnormal development or disease, which is almost the same thing in many ways, all right? So the signal modulation, the factors that modulate the signal in notch are extremely, extremely important. And that is the essence of the problem that we're looking at, okay? Now, one thing that, and I will be talking a little bit about evolution, not in any sophisticated way, but I'll touch upon that. Now, if you, one thing that we have learned over the years is that the signaling mechanism, the signaling pathways that control whether you're a fish, a prime minister, or a fly, seem to be the same. So the signaling framework that is basically able to build a three-dimensional organism is the same. A remarkable fact, unexpected in many, many ways, all right? So and I think that, of course, begs the question of how is it possible for evolution, if they are using again and again the same cellular framework, to invent novelty. I mean, we're definitely not the same with flies, at least most of us. But so you have to, in order to evolve and invent novelty, you must be doing something. And I think for me, this is the problem. That's the problem that bugs me. And I think that I'm too old to hope to solve it. But I'll try. I'm trying. So let me go back here and tell you a little bit more about tweaking of the signal, all right? Because that's, as I said, this is crucial. If you tweak the signal, and if you can trigger cancer or not, and so on, all right? So I'll give you some examples, and then we'll go into the more general aspects of it. One example is Deltex, is a ubiquitin ligase that happens to interact physically with notch. And Kurtz is another molecule that interacts with Deltex, all right? So now if we take Deltex and express it in the margin of the cell, of the wing is normal. If we take Kurtz and do the same, the wing is normal. If, on the other hand, we have notch, Deltex, and Kurtz, then you see the loss of function phenotype. Now you all know that the loss of function phenotype means not swinging in our systems, in our experimental system. So what happens in vivo, all right? If you look at if you make a mosaic with the magic of genetics, and that's, as both Iva and Gary said, genetics is like playing the piano if you know how to play it. I mean, it's absolutely beautiful. So if you make a piece of tissue that does not have Kurtz, this is the dark piece right here. So these cells don't express Kurtz. And if you ask what happens to the Notch protein, you will see that the Notch protein here is elevated in those. So absence of Kurtz elevates the protein. Does the protein signal? And if you take a signaling marker, you will see that in spite of the fact that we just learned that this empty Kurtz minus patch has elevated Notch protein, you can see that the signal is only just in one part of it. Emphasizing context. So you may tweak the locus, you may tweak the signal, but it depends on the context whether you will signal or not. reminding you again that this is extremely important because of disease again, all right? So if you have Kurtz there, it can actually be a modulator, like a dialing up or down, depending where you are, Notch signal. And if Notch signal is important for disease, 10 minutes more? You are telling me that I have already spent 10 minutes. Or that I have left 10 minutes. I see. Thank you. I don't speak English, I'm sorry. Yeah, okay. What if we were to look at, doing again genetics, to look at modifiers of this, I'll respect that, okay, I'll try. Yeah. If you ask the question now, how can I find modulators of this activity? So Notch, delta, Kurtz, you can see the wing notching. Can I find modulators doing the magic of genetics? The answer is yes. I can find suppressors, I can find enhancers, over 100 of those. And if you look at what are these proteins doing, they are doing all kinds of things. There's no logic. They are not phosphatases or ubiquitinases or whatever. It's all over the place. So that's not a good idea. I mean, to try and figure out in the mechanistic terms that Gary, for example, outlined, to find out exactly what's happening. It's very difficult. So how complex is the genetic security underlying Notch? And what happens with integration? We'll do another experiment. We will knock Notch activity in the wing margin and look for the genome, start mutating every single gene in the genome, all right, or 50% in this particular case, and ask the question, can I find, again, enhancers and suppressors? And the answer is yes, I can find enhancers and I can find suppressors, but if you ask how many, in this particular context, there are 500 of those, or more than 500. Absurdly complex, all right? Integration. Notch E5 and mirror are two genes that came out of the screen. Now, Notch is, Akits-Cute is a known target of Notch. Mirror is also affecting Akits-Cute. And Orocan and Copolitan, who are negatively controlled by Notch, are also affecting Akits-Cute. Wingless can actually also negatively influence Notch. TGF-beta can positively influence mirror in this particular case. And hedgehog can positively influence mirror. And finally, E5 can actually positively influence TGF-beta. So you can see that the integration of these pathways is profound and confounding, both, trying to isolate, to understand it molecularly. Now, I will, and again, I'm reminding you that if you tweak the signal, you can get disease, you can get cancer. So I'll show you one example, again, of integration and of how important it is to tweak the signal. This is a gut, of a mouse gut in this case, and we are activating Notch in the crypts. And you can see this is a proliferation marker. Cells proliferate. If, however, you inactivate wingless, this proliferation doesn't work. I mean, it doesn't proliferate anymore. So Notch stimulates proliferation, but only in the presence of wingless signaling. Again, integration if you look at the gut of a mouse that is heterozygous for APC, that's why we do colonoscopies. There are 5 lesions per mouse gut that are formed with APC plus, APC over plus, and heterozygous. And if you activate in those Notch, you can get 115 lesions. And Kaplan-Meier says the same thing. Notch by itself, the green is okay over this period of time. APC over plus, eventually they develop adenocarcinomas. But if you activate Notch, the mice die quite fast, all right? Now, I will skip. How much time do I have? You're making noises, 5 minutes, okay, all right. So what I just showed you, though, is that Notch per se, in this context, is not oncogenic. But it needs something else. It needs another X, wingless in this particular case, to become oncogenic, all right? So can we identify this X? Is there any way of identifying X's? Again, mutations and genetics. This is a normal eye. This is an eye that has activated Notch, so it's slightly bigger. And these are, you can see now that you can find enhancers that are exploding the eye in terms of proliferation. And you don't have to be an expert in eye development to see that, okay? Again, the numbers are above 100 of these genes that, and all over the place in terms of ontogeny. Doesn't make any sense. If you look at all the, if you combine the literature as well as our screens in terms of looking for modifiers of Notch, the answer is confounding again and flabbergasting. We call it the Notchome, and there are more than 1,500 genes that have been associated one way or the other with Notch modulation. All right? So this is really a headache. I mean, how do you address that? The means to address some complexity of that kind are becoming available, and it is very, very exciting about that. However, what we decided to do about almost a decade ago is start looking at protein-protein interactions to try and somehow put a foot in the door of trying to understand the underlying interactions. And so the approach, cDNA clones, you tag them, you express them in cells, and then you analyze, you fish out the tagged clone with everything that sticks to it, and you analyze by mass spec. And the mass spec actually analysis has revolutionized the way things are being done. And then you make a map of these interactions, which I will go into excruciating detail in the next few days. So in this particular case, colors mean ontogeny-related genes. We identified, we did about 8,000 baits and got this map. Now, how useful and what's the point of the map? If we look at a closer view of the map, you will see that these are defined complexes. So there are several questions that we are asking, and I will not go into any kind of detail. First of all, do complexes have a predictive value? Does that have any predictive value? The answer is yes, to a large degree. Gee, this is like a Howard Hughes meeting where at the end of the day they cut your head off if you will. Yeah. So do you distracted me. Do complexes have a predictive value? And the answer is yes, to some degree, not 100%. What happens if you destroy a complex in one or two proteins? What happens to the rest of the complex? Is it affected or not? If you eliminate now a complex, one way or another, how does it affect the network? So next door neighbors, I mean first degree or second degree neighbors. Very, very interesting questions. And what happens if when one connection in a node, like these things here, that you have connections between complexes if you destroy that connection? Can you destroy the complexes and to what degree? Now, what happens when protein quantities are altered? And we're doing now isobaric experiments, because genetics, sometimes they do, address quantity, but not very often, all right? And of course, a big important is AlphaFold has given us delicious possibilities thanks to Demis Hasabi and his colleagues. I mean, absolutely amazing possibilities. And finally, conservation. We heard by a number of people, by Iva and Gary specifically, that if you maintain conservation across species barriers is an indication of importance, of biological importance. So we collaborated, I mean, they did most of the work, mind you, with my colleagues Steve Gigi and Wade Harper, and did a proteome of a human proteome on the left, as you can see. So now we can do a number of things, and they are a lot of fun to do and very much important to do. Does a complex in flies, is it conserved in humans? Again, all these relationships that I indicated before, how are they compared across species? A crucial questions, really. All right, and finally, one thing that is for me a very important problem is whether this integration that we can identify in flies, is that integration valid in humans? You have, I told you about knots and proliferation and winglets in the middle, all right? Does that hold in flies, in humans, in worms, or whatever? Because conservation is important. With this, I will thank you very much for your attention, and I relieve you to go to lunch or something. Oh, yeah. Thank you. No, it's okay. It's okay. It's good. It builds character. He says it builds character. Okay, can I get the microphone back? Thank you. So really, a marathon of amazing science. I guess that's what I would call today. A fantastic day, incredible talks, incredible laureates as ever in this in the Gairdner Awards. It's really a remarkable morning of insights. Everybody spoke about collaboration and teamwork. It's everybody talked about conservation of mechanisms across evolution. Everybody talked about how understanding fundamental science gives you insights into disease and how to treat disease. So everything interconnects, and that's one of the great messages. So I am going to thank you all for staying with us, and I hope everybody enjoyed everything that you heard. I certainly did. Your engagement really makes this a special day as well. We are going to continue the celebrations. Some of you who are here today will be with us celebrating at the ROM tonight in the gala. Until then, please take away all the inspiration you heard. And I will also ask the laureates, you're going to lunch with some students. The students who are going to the lunches, but only the students who are going to the lunches, please pick up a box outside, a lunch box, and go up the stairs. We have the tables where the laureates will join you for lunch. So everybody else, thank you very much, and enjoy the rest of your day.